It's my pleasure to introduce Eric Alm, our third keynote speaker for this workshop. Eric came originally he did his undergraduate at University of Illinois, Urbana-Champaign, and a master's degree at the University of California in Riverside, with a PhD from the University of Washington in Seattle, and finished up his training at Berkeley. Eric is the professor of biological engineering at MIT and co-director of the Center for Microbiome Therapeutics and Informatics. And in 2013, his laboratory launched the nonprofit OpenBiome to meet the clinical demand for fecal transplantation. And as of the writing of this bio, the treatments that OpenBiome has sent out have treated over 25,000 patients. So his research is primarily focused on translating the microbiome research into the clinic. I heard Eric speak in February at a Keystone meeting, and I thought it was fantastic. So with that, further ado, I'd like to introduce Eric Alm from MIT, whose talk is on microbiome interventions from fecal transplants to synthetic microbial therapeutics.
Well, thanks, Ryan. Thanks for everybody who's here. So a real special treat and quite an honor to be included in the lineup at this workshop. When I started at MIT about 11 years ago, the one thing I said that I would not do is get involved in biomedical research. I was an environmental microbiologist, and I said there's enough people doing biomedical stuff. Microbial ecologists aren't needed, right? There's already enough people working on it. And I got drawn in by a colleague who was working on the Human Microbiome Project and wanted help with statistics. And I said, well, statistics sounds pretty safe. That doesn't sound biomedical at all. And I started working on it, and it was really so exciting. I've kind of changed my entire direction, and that's sort of all we do now. So in many ways, it's a special honor and a special treat for me to speak here about the HMP, because it was the HMP that really ended up directing in a large way my ongoing research program. So folks who've seen me talk before have seen this slide, and this is the first project that I worked on in the human microbiome. This is when I sampled my own microbiome, gut microbiome, every day, along with a really talented graduate student, Lawrence David, who's now an assistant professor over at Duke. And on the top is my microbiome over the course of a year, and on the bottom is Lawrence's microbiome. And if we dive a little deeper into my microbiome, one of the sort of interesting things, there are interesting things that happened to both me and Lawrence during the study. One of the interesting things that happened to me was the salmonella infection. So I got food poisoning after ordering some undercooked French toast about 3/4 of the way through the study, and I got salmonella. I didn't get antibiotics, so I got to see the infection run its course. And one of the really interesting things that happened, and so on this plot, the rows are the different bacteria. The warmer colors indicate there's more of that bacteria on that particular day. Cooler colors indicate there's less. And you can see that once I got salmonella, all of these bacteria who had been the major players in my microbiome up until that point sort of went away. And most of them never came back, okay? And they were replaced with these bacteria down here, some of which were brand new and were never seen in the 170-odd days that we had sampled up to that point. And some were present, but they were at very low abundance and never able to really get a foothold in whatever niche they were supposed to populate. And this really got me thinking, if the microbiome can be reprogrammed like that, and we can figure out what is the difference between a healthy and a sick microbiome, then maybe we can go in and we can use this clinically, right? We can fix a broken microbiome by putting new strains in. One of the things that kind of reinforced that belief is these bacteria up here that went away and these bacteria that replaced them were very, very similar. They were very closely related to each other. And so here's sort of an overview of the tree of life for the bacteria that were living in my gut. And the blue is sort of the blue from the previous plot. So these were the microbes that basically went extinct in my gut. And the orange ones were the ones that replaced them. So for each bacteria that left, it was replaced with another bacteria that was very similar, right? And so that the sort of functional capabilities of the microbiome didn't really change very much. And the other thing that I want you to notice, and I'll come back to this later, is that all of these bacteria that initially went almost completely extinct, except for these little white spots here, and then were almost completely replaced, were a subset of the firmicutes, okay? And I'll come back later and I'll show you functionally what that subset actually is. Okay, before we get there, one of the next projects I got involved in, and this kind of follows directly from this idea that, hey, maybe we can engineer the microbiome, is the idea of engineering transmission and using fecal transplants for Clostridium difficile infection. And in some ways, C. diff is certainly antibiotic usage is a very strong risk factor for C. diff. And the way I think about it is antibiotic usage is sort of our crude attempt at engineering the microbiome, and it's an attempt perhaps gone awry because then it leads to this higher risk for infectious disease afterwards. So can we try other methods of engineering? And so oftentimes we have this healthy microbial gut ecosystem, apply antibiotics. It's like clear-cutting that healthy ecosystem. We get invasive species like C. difficile, and we can sort of get caught in a cycle here, right? And if we go through the cycle 3 times, now our C. diff infection is a recurrent C. diff infection, and we're allowed by FDA under enforcement discretion to treat it using fecal transplant. That's sort of what we do at OpenBiome. And I got involved in a study, an FMT study run by Libby Homan over at MGH, and together with Dirk Jeevers and Ramnik Xavier at the Broad, collected genomic data, and in particular metagenomic data on this cohort. So we had 4 healthy donors treating 20 patients with about a 90% success rate, okay? And importantly, at least one of our donors treated 12 patients, so we got to see this sort of in-human experiment repeated multiple times. We got to use the data to build models. And one of the models that we wanted to look at, this is work done by Chris Smiley, a really terrific graduate student in my lab. One of the problems that we wanted to solve and model is if we look at the strains that are in a microbiome, and so this is a C. diff patient, typical C. diff patient before fecal transplant. Here is our healthy donor. Okay, so you can see those profiles are pretty different. I'm not showing you all the different names of the bugs, and they're in no particular order, but darker colors means there's more of that bacteria in that particular sample. This is what our patient looks like after a fecal transplant. So we stared at that and we said, gosh, it doesn't look like there's any very simple rules. It doesn't look like it's, A plus B equals the combination. It doesn't seem to be like an AND gate or an OR gate or any of the simple rules that we can kind of put together. But Chris wanted to see if he could use machine learning to build a model that would predict whether or not, A, sort of whether or not a species would engraft, and B, if it did engraft from the donor to the patient, what would its abundance be? And when we start to think about designing synthetic microbial therapeutics in the future, these would be getting away from something as crude as a fecal transplant and taking bugs in pure culture, feeding them to patients, and hoping that they engraft. Well, for a lot of the mechanisms that we care about, we need them to be abundant. We randomly pick a bacterial species from the human microbiome, they're enriched at the low end of the curve in terms of abundance. So your average species is not going to be very abundant at all. And if you want your species to, let's say, produce a commodity good like butyrate or something like that, if it's present at 0.0001% of your total biomass, it's not going to be a very competitive producer. It's not going to alter the, the overall pool of butyrate very much, right? So predicting abundance is important, and what we found is that we could predict whether things engrafted quite well, and we could also predict abundance. And importantly, if we say, hey, here's our cutoff, anything lower than this we don't want to use in a synthetic therapeutic, if we picked all of these strains, for the most part, we would get things that actually were quite abundant in the patient. And so because we can do this, it sort of paves the road from going from fecal transplants forward to more synthetic treatments. So we built the model. This is what it looks like on the data set that I just showed you. A lot of the things that seemed hard to predict here, the model actually gets right, and it gets a few things wrong. And it gets a few things right that are very surprising. The first time I gave this talk, I was shocked by my own data because I hadn't looked at it that closely, and I said, oh my God, there's an error. And someone in the audience was kind enough to point it out to me. But we've gone and we've looked, and if you look at this species right here, it's not present in the patient and it's not present in the donor, and yet it's present after the FMT. The surprising thing was it was predicted by the model. And it was predicted by the model because here's a strain that is rarely present in our patients or donors, but often shows up in patients after fecal transplants, and the model is able to pull that out. So there's these species that play an important role potentially in succession and things like that. And these are things that we're sort of learning by mining these data. So that was all at generally the OTU level. We wanted to look a little bit closer at the species level and even drill down to the strain level. So Chris wrote a program called Strain Finder, and what Strain Finder does is it takes metagenomic data, which are these very short reads, and then tries to infer what is the strain content. And if you apply Strain Finder to a single sample, it's actually pretty good at telling you how many strains there are. It can't tell you what is the genotype of those strains, because there's not enough information. But if your number of samples is, generally speaking, your number of samples is greater than your number of different strains in those samples, okay, so you have strains appearing across multiple samples, then the statistics work out such that you can often predict not only how many strains there are and how abundant they are in each individual sample, but what the genotype is for each of those strains. And here's just an example of Strain Finder. And so here we have different numbers of samples, from 2 samples all the way up to 64, and from 2 strains all the way up to 32 strains. Okay, so we're not doing very good. So the blue colors are better, like dark blue is essentially a perfect prediction. But we're getting very good predictions across most of the combinations of different numbers of samples and strains. And here's some example data where red is the predicted genotype of the strain, and black is the true genotype of the strain. And you can see that for every true strain, there is a predicted strain that matches it very closely. Okay, so we applied this to the same study, the fecal transplant study. And so here we're looking at strains in a Ruminococcus species, and here we have our donor number 1 has 2 strains. Okay, the blue one is about twice as abundant as the green one. It goes into a donor over here and 3 different time points. And you can see that both of the strains here transferred over to the patient. And this was a case where the donor had this particular OTU, and the recipient, the patient, did not. And so the strains get transferred over. Here the donor has 4 different strains, and those strains get transferred over as well, but there's a lot more fluctuation in their relative abundance. Here's F. prausnitzii, and this was a case where the donor had 4 strains. The recipient picked up those 4 strains, but then even though that recipient had never had F. prau prior to the fecal transplant, once they got FPRAU, they were able to acquire new strains, this red strain here, from somewhere, from the environment or something maybe very, very low abundance initially that was able to thrive once there was sort of a critical mass of similar strains of the same species. And this is something that we saw over and over again. So we have these strains of donor origin, and then these strains that we call environmental because they're from some other source. Another case was where the donor had a strain and the recipient also had a strain of the same OTU. And some of the times, we would see that pre-FMT and post-FMT didn't change very much. So these donor strains were not really able to get much of a foothold in the patient. And sometimes we saw the opposite behavior. So after fecal transplant, the patient looked a lot more like the donor at the strain level. So we saw that this competition can happen. And at the end, I'll tell you a little bit about some of the rules that we're starting to discover about what affects that competition. The other interesting thing we noticed, and you kind of saw this on the diagrams already, if a strain engrafts in the patient, and only about 30% engraft in the patient, but when they do, the patient gets essentially 100% of the strains that were in the donor. Very rarely do they only pick up some of the strains. So it's sort of this all or nothing behavior. Most of the time, they, given OTU doesn't engraft, but when it does, all the different strains engraft. And you can see some examples of that here. Even the really quite complex communities seem to engraft, and all the different members come across. And this is surprising since, in general, if you look at any one of those strains, small probability that they engraft. But if you know that they're closely related bugs engraft, then there's a high probability. So there's definitely a sort of interaction there. This is kind of an overview, and the only thing I want you to take away from this slide is, this is for all the different fecal transplants that we looked at, can we assign the strains to being either from the patient initially, from the donor, or strains that were never seen in either, and those are what we're calling environmental. And so the environmental is shown in blue, and you can see it's a significant portion. It's almost a third of all the strains. They can't engraft until the fecal transplant, and they need some of the donor strains there to make the environment favorable, but once the strains are there, then these environmental strains can get a foothold. So about a year and a half ago, we got a grant from the Broad Institute to dive a little bit deeper and really investigate all these differences between strains. And so what we did is we took the open biome donors, took the collection, and started to generate isolates. We got about 7,000 isolates, whole genome sequences for them, metagenomic time series, 16S time series, and metabolomics to really characterize in a lot more detail what are the phenotypes and genotypes of these strains. Sean Gibbons is a really talented postdoc who led up this project, and this was his sort of sample plan for 16S and metagenomics. So from these donors, they come in about 4 times per week. And so for a number of them, we were able to get very long time series up to 2 years, and then follow it with 16S and metagenomics. So the advantage of this is now we have these. We have them stored, and we can revive them. And if folks are interested in particular strains, we're hoping to be able to make those widely available as soon as we kind of hire somebody to maintain the collection and start mailing it out. But these are going to be very well characterized collection that we hope is a valuable resource to the research community. It's pretty comprehensive. So if we sort of weight the different strains by their relative biomass, you can see that we actually have very good coverage. So the dark parts of the pie here are the strains, the percentage of those strains that we have in the freezer from 11 different donors, and the shaded parts, these are the strains that we don't yet have. And so you can see we have very good coverage, and the ones we don't have, it's because they're not very abundant, and it says that we're not fundamentally missing anything in our approach. We just need to do a lot more isolations because they're less frequent. Some of these include strains that have been difficult to culture previously. So human isolates of Akkermansia, at least to my knowledge, there's really only one that was isolated about 12 years ago. Now we have dozens kind of in the freezers, and hope that that's going to be pretty useful. And so we have other species beyond Akkermansia mucinophila that are of human origin. Teresibacter, again, as far as I know, there's just one human isolate, and we have over 500 of these, and we know from culture-independent surveys that they're a fairly important part of the spore-forming fraction of the microbiome. So I want to dive a little bit deeper on what we're starting to do with this collection. So here we're looking at 12 different donors, and we're looking at B. fragilis. And so what you can see is that B. fragilis in each of these donors, I haven't put the trees together on one large tree because the structure is very simple. All of the genetic diversity in B. fragilis within each donor is clustered within that donor. And there's a relatively small, at most dozens of mutations from the last common ancestor to each of the isolates. And so we're fairly certain that all the diversity in B. fragilis in any given person evolved during the course of that person's lifetime. So that person was inoculated by one strain. Maybe they were inoculated by multiple strains, and only one sort of outcompeted the others. But when we look at these isolates, we see that there are distinct trees, one for each of the donors. And so what we did is we kind of did a deep dive on B. fragilis, cultured a number of isolates from individuals, did metagenomics, gene sequencing to infer whole genome sequencing to get phylogeny, and then metagenome sequencing was already done for this library, so we could go back and see with sort of daily resolution how these different strains were competing with each other. So here's an example. So this is again our B. fragilis, and this is the distance to the most recent common ancestor in terms of SNPs. And you can see some of them, had like up to 10 SNPs. Some only had a few. So these might have those individuals might have been inoculated with that particular bug more recently. Okay. And this is one donor 44 that we had the extremely long 2-year time series for. So that worked out pretty well, and I'll show you some of the dynamics there. Donor 440 was pretty interesting because it actually had the highest number of SNPs from the isolates to the most recent common ancestor. But when we looked at the tree, all the SNPs were on one branch of the tree. And when we started to look at what type of SNPs those are, we saw that they were very highly enriched in transitions and even a specific type of transitions. And it really looks like maybe a defect in error correction, and we essentially have a hypermutator that evolved hypermutation within the human host. Well, the thing that we're most interested in, of course, was not looking at the transition-transversion ratio, but trying to identify which of these point mutations that were occurring were due to positive selection. And so it's a relatively small number of strains, so it's difficult to use any of the specific kind of PopGen type tests for positive selection. But what we reasoned is that if we start to see multiple hits in the same gene or in a similar gene in the same pathway, that gives us some confidence that the reason that we're seeing excess hits in the same gene is because it's conferring a selective advantage. And so if we look at all of our SNPs, the SNPs that separate kind of the strains in one person from the strains in another person, we see that the DNDS ratio is quite low. It's what you expect for SNPs that differentiate two different species. So, many more non-synonymous, many more synonymous mutations than non-synonymous. On the other hand, if we look at strains that where we get multiple hits in the same gene, or multiple hits in the same gene but in a different strain, maybe even in a different person, and then we look at the DNDS ratio, we see that they're mostly non-synonymous. And this really was kind of the smoking gun for us that, yes, there's a qualitative difference here, and when genes are hit multiple times, they tend to be enriched in these non-synonymous hits. Suggesting positive selection. The other analysis that we did is they're also highly enriched if they're hit multiple times in membrane protein genes. So this is a list just for BFRAG of the genes that kind of crossed our threshold as being pretty interesting. And you can see there's a lot of things that are perhaps not surprising. So capsule polysaccharide biosynthesis and some transcriptional regulators and sensors. But the thing that really grabbed us was these Sus family proteins. We heard a great talk yesterday on sort of what some of these Sus proteins are doing. And we see that this, at least in B. fragilis, this is just the single, class of genes or pathway that's absolutely the most enriched in within person evolution. And so what you can see here, all the red dots are the non-synonymous, and so there's only one synonymous mutation. These are different strains, the rows, and the columns are different people, and the X's are the genes being lost. So there's a lot of gene gain, wholesale gene gain and gene loss, in addition to accumulation of point mutations. And I'll get to that in just a minute. We can look at where these mutations are occurring in the three-dimensional structure, and they seem to be occurring in the interface region between Sus-C and Sus-D, and they're also occurring around the hinge region, and that makes a lot of sense because those would be two pretty important regions for gene function. We don't yet know if, kind of following yesterday's talk, are these SNPs actually, helping different people more efficiently use, let's say, different types of starch, something like that? Are they adapting to an individual person's diet post-colonization? Or are these genes so important and they're expressed so highly on the cell surface that they're a good target for phage? And they're constantly evolving to sort of outwit the phage that are evolving to kill them. And so we have a number of experiments going on now to sort of flesh out this question, and it may turn out that it's a little bit of both. So now we'll dive into one donor. This is our donor where we had about 2 years worth of metagenomic samples, and so here are all the isolates from that donor. Okay, and on the phylogenetic tree, I've just colored where the different mutations happen. I've colored it by branch. And we can go to the metagenomic data, and we can track each of these SNPs, and we can see that they have a dynamic pattern, and we can see that these two mutations came up relatively late, probably during the course of our study, and then they grew and they really took over, but they only took over one of these branches. And so because we have so few SNPs, we can build a perfect parsimony tree, and then we can map those SNPs, and we can really build a model of, even though we're starting with metagenomic data, we don't have whole genomes, we can make very good inferences about what all the strains are and what their abundance is at any point in time. And so we can only make kind of general inferences about what happened in the years predating our sampling, but during our sampling, we can build a fairly precise model of how much of each strain we see at a different time. We can see sometimes we get mutations that crop up and then die off over here, and sometimes we get mutations that crop up and really take over, but they only take over sort of one of the arms, and we have this sort of constant coexistence of the two different strain types that I showed you in the previous slide. Okay. So we've talked a lot about point mutation. One thing my group is always really interested in is horizontal gene transfer, and we got inspired by this study on Kzymes, which showed that most likely in the Japanese microbiome sort of mobile gene pool, there are these genes to digest porphyrin, which is the polysaccharide that makes up these nori sushi wrappers. And it was horizontally acquired from a marine bacteria that was feeding on the seaweed. So we built this, and this was really the first ever kind of map of horizontal gene transfer and sort of hotspots. And the thing we found, even though we included all the different genes, all the different genomes in GenBank at the time, and we had a lot of non-human sort of environments, we saw that the human microbiome was the real hotspot of horizontal gene transfer. It's where we saw most of our hits. I was fortunate enough to work with a really terrific postdoc, Alana Brito, who's now an assistant professor at Cornell, who took this study out to some rural villages in Fiji, where she collected samples from about 300 different individuals, came back and analyzed those for community structure and gene transfer. And what she found is that although the HMP metagenomes kind of all clustered together and the Fiji metagenomes clustered together, when she looked within villages, they were essentially on top of each other. And one of the things that was causing the metagenomes to cluster together was that there are these population specific genes that were, we can see some over here. These are mobile genes, but they're specific to the HMP, and they're rarely found in Fijians. And then here we have mobile genes that are highly enriched, but specific to Fijians and rarely found in the HMP. And we can look at what those genes are, and they tend to be enriched in these glycoside hydrolases that allow their host to access different parts of the diet. And they eat very different starches. And we can see a number that are kind of enriched in the HMP versus Fiji, and a number that are enriched in Fiji versus HMP. Okay, so we initially published the paper on horizontal gene transfer. It was back in 2012, and I got this email from Yara Oren, who I thought was a postdoc, and she demanded to have a Skype with me. I said okay, and she told me how I had gotten everything wrong, and I had missed the bulk of the signal in my data because we wanted to be really conservative about what we called horizontal gene transfer. We had this 500 base pair cutoff. If it wasn't at least 500 base pairs, we didn't look at it. And she said, that caused you to miss the most exciting part of the story. And the most exciting part is that even for genes that rarely ever get horizontally transferred, their upstream promoter regions, their regulatory information often gets transferred. But it's usually less than 500 base pairs. So she came out to my lab, finished the project with me, and coined this term HRT, which is horizontal regulatory transfer, and identified that as a key type of regulatory switching that bugs use to adapt to their environment. And so here you can see, here's a MET-R gene, okay, this is the regulator. All the E. coli's kind of grouped together, the Enterobacter is grouped together. Here's MET-E, we have the Enterobacter and the E. coli's. But when we look at the promoter region in between MET-E and MET-R, what we see is that the Enterobacter is over here with the E. coli's. And so what it looks like is the E. coli has picked up horizontally transferred regulatory region from Enterobacter. And so she went on to show that if you look at E. coli strains that have picked up that have switched their regulation versus in two different strains, it affects their function. So it affects their gene expression. So if they've picked up a new regulatory region, they're much more likely to have divergent expression across two different strains. And at least for the MET-R, MET-E example, she was able to show in the laboratory that it actually had an effect on gene fitness. And she also showed that this was pretty widespread across the tree of life, and it was also species dependent. And so we see very different rates of regulatory transfer and regulatory switching across different species, and we still don't really understand what the rules are there, because it doesn't correlate very obviously with the overall amount of recombination in those species. Okay, so getting back to kind of the main arc of the story, we wanted to look, and so when I showed you these maps, and I'm showing you all this stuff about horizontal regulatory transfer, all of these things are happening over vast timescales. And so for DNA to be 1,000 base pairs in 99% identical, well, that means you could have had a horizontal transfer yesterday or last week. You could have also had a horizontal transfer 5,000 years ago, sort of the dawn of modern civilization. Those would both get you in that 99% regime over a single gene. So one of the key questions to me was, well, when we get an OTU and we get colonized by a new species, can that species pick up genes that are maybe in other bugs within our microbiome? Because that would mean that we're missing a lot when we get a 16S, and we can't just look at 16S and reference genomes and get the whole story, because the fundamental functional properties and metabolic capabilities of these bugs are adapting to our own environments via this very efficient path of horizontal transfer. And I spent a long time looking at single-cell genomics and a lot of things to try to find evidence of this transfer. And I'll walk through, I think we finally found it, and I'll walk through some of the evidence based on the isolate genomes that we now have. And so this is a call it ME2. It's a prophage, and here we can see the dark colors here are just sort of the coverage in metagenomes that correspond to these isolates. And what we see is that these isolates form a clade. Okay. All of these are descended from a single bacterium, probably no more than a decade or two, that colonized this person no more than a decade or two ago. And this subclade right here, they pretty much all have this prophage, except for this one that has likely lost it. So to me that implies an acquisition, although you could argue it could be perhaps multiple losses. But if we go back to the genomic data, what we see is that on day 0, okay, none of our, so we have isolates from day 0. None of our isolates have the prophage, but our metagenome has the genes. If we go to day 340, our isolates have on average sort of 1 copy of the prophage. And then we also see it in the metagenome, and we've done this for other genes as well. Okay, and so the important thing to notice here is when we look at the isolates and we sort of normalize everything to 1, we see that there are more copies of this phage in that metagenome than what we would expect based on the isolates. And we've gone back now and we've confirmed that we can find some of these elements in other species within that microbiome, and those other species kind of explain the difference here. So we think we've got a number of cases where we really have that smoking gun microbes coming into a genome and then picking up new functionalities within the course of a human lifetime. So I talked a lot about bFrag. Other bugs may have different dynamics. And so here we're looking at E. coli. And this is our day 0 time point. We just have one isolate. We look at day 174. Now we see a few bugs that look like this, and also we see it splitting here. When we get to day 372, all of these other bacteria that we saw on day 174 are gone, and we only see bacteria from this particular clade. And we look at day 709, kind of all of those bugs are gone, and we see these two new clades. And so unlike bFrag, where we're accumulating genetic diversity over decades, with E. coli the story is a lot different, and you come back a few months later and a lot of the previous standing diversity is lost, and so likely has something to do with the particular niche and lifestyle of that organism. So the last thing I want to talk about is endospores, and this is something that we all kind of know is going on, and it's a sense, well, we just saw that species can, change their fitness by acquiring point mutations and deletions. They can change their functionality by acquiring new genes. Well, here, here's a case where they can change their morphology just by making a sort of regulatory choice. And so what Sean Kearney, a terrific graduate student in my lab, did is he took whole community and then he processed it to kill everything except for endospores, extracted, sequenced both of those communities, just standard 16S sequencing, and then compares them to identify which are the bugs in any particular sample that are enriched in spores. And he did this for dozens of people, and then also through time series, so we can see of the potentially spore-forming bugs, how often do they, what percentage of those bugs are present as vegetative cells versus spores at different points in time. And so, here's what those data look like. Here's the bulk community. Here's after enriching for endospores. And so you take a lot of this diversity, and a lot of it goes away, and you're left with just a few clades. A few clades of Clostridia, and this guy I won't even try to pronounce, and bacilli, and things that are known to form spores. We also always see actinobacteria. And so even though we know they don't make conventional endospores, we can isolate them from stool, and they're at least as hardy as normal spores, and so we think they have an alternative kind of dormant state that functions very much like a spore. I don't have a timer, so looks like Ryan is telling me to. Okay. Yeah, we're about done. So we looked at these spores, and we went back and we looked at the dynamics over time. And what we found is that in most of the time series we have two different groups, okay? And at least one of those groups is highly correlated and also highly variable over time. And what we found is the vast majority of our endospores were in this highly variable component, along with Bilophila. And that was interesting to us because we know that bile acids are probably required for spore germination. And so we took a number of our strains and kind of tested this. And right at physiologic concentrations is when we start to see things moving from 100% spore to much lower fractions of spores, so mostly vegetative cells. And the other interesting thing we found about these spores is if we looked at how many different people share spores, for bacteria where almost 24 out of 24 people share that bacteria, they were generally spore-forming bacteria, whereas for non-spore-forming bacteria, most of the time you will only see that particular OTU in a subset of your population. And so what this led us to think is maybe there's a trade-off between being highly variable and not very permanent, but also being able to transmit really well across multiple people. And so if we go back to the time series that I showed you initially, right, we can look at spore formers and non-spore formers that were abundant initially, and we see that switch post-infection. So in that post-infection regime, I get colonized by a large number of spore formers. And in fact, all of these bugs down here that I noted at the beginning of the talk, these are exactly the spore formers that we're talking about. So it's the spore formers that got wiped out as a result of my salmonella infection, and it was spore formers that very rapidly replaced them. We can look at it in the context of C. diff, and what we can see is that the ratio of spore formers in healthy controls versus first-time C. diff infection versus recurrent C. diff infection is gradually lower and lower. And we can also go back to the story I told you about fecal transplants, where sometimes the pre-FMT recipient strains win out, and sometimes the donor strains win out. And a lot of this is explained by spores. So for spores, which are these resistant OTUs, we mostly see donor strains when this happens. So if the patient and the donor both have a particular strain, if it's a spore former, we'll almost always see the donor strain, whereas if it's not a spore former, we'll often see the patient strains. Okay, so I think I'll end there, and thanks for the invitation, and thanks for your attention.
So I think we have time for at least one or two questions for Eric.
Nice talk, Eric. My question goes to the point that many of us try to define a healthy microbiome. And what I think your work suggests is maybe we should rethink how we do this. That is, maybe we shouldn't be thinking along the lines of species and strains, but we should be thinking along the lines of actually defining functional subsystems or genomic elements that are essential for health. And in that regard, in thinking about what we can do in developing tools to achieve that, I mean, I found your prophage findings very interesting, and that could potentially be a way of introducing genomic elements in a non-stochastic way and trying to achieve that nirvana of a healthy microbiome. I was wondering what your thoughts on that were.
Yeah, no, I think it's a good point, and I think there are certainly some functions that we're going to find are highly variable and maybe can be acquired, when you get to a new host and when you need them. And I think there's going to be other functions that are going to be kind of more deeply embedded in metabolism and more difficult to transfer. And so I think what we need to do is just kind of look at these on a phylogeny and identify, okay, which are the functions where if I know the phylogeny, like my prediction is probably fairly good, and which are the ones that are going to be very difficult to do that way. Looking at BFRAG, we're just starting to look at other groups, and I think it'll be interesting to see what are the selective pressures when an organism, enters a new host. And, a lot of organisms don't have the Sus family gene, so they're doing something different. Is it always kind of carbohydrate usage? Is it something more maybe mundane or not mundane but less useful, like just knowing that they're evading phage? I don't know.
Hi, Eric. Liping Zhao. So for the environmental strains that you term, is it possible that they are actually in the rare biosphere of the donor's microbiota? And the related question is, how many strains in a person's gut microbiota can you estimate? Are there any rough numbers?
Yeah, so in terms of how many strains we can estimate in any one person, It might depend a little bit on how deeply you sequence, to be able to see things that are very, very low abundance. But, for things that are at least a few percent, is not, generally not more than like 5 or so. Now, I showed you this data on bFrag where we have like 30 different like strains, but most of those differ by only one SNP. And so if you run Strain Finder on, those like metagenomic time series and kind of see how it pans out, it will say there's 2 strains, one corresponding to each of those groups that, are about 10 SNPs apart.
About the environmental...
Sorry, yeah. So the environmental, I think it's unlikely that they are, although, totally possible that they're low-level present in the donor. It's more likely that they're low-level present in the recipient. And the reason I say that is for most of these, we go back, we look at the donor, but then we look at all like the other 11 patients that were treated by that donor, and none of them, if the donor doesn't have a single read that matches it, and none of the other 11 patients had a single read that matched it, then it seems more likely that it was in the patient, although maybe they're both just so far below our detection limit that you wouldn't see them. So I know people get upset when I say environmental, but it's more of a like from my perspective, if I'm thinking about designing a synthetic microbial therapeutic, I don't really care the origin, like everything came from somewhere. The important thing is I'm going to put this isolate, and maybe it's a genetically engineered organism to combat inflammation or something like into a patient, and once it's there, it may be outcompeted by things that were never in that patient. And I think that's sort of the take-home message from me on what the important consideration is regarding these environmental strains, right? And also the fact that, well, with horizontal transfer and just accumulation of SNPs, function can change. So it's not enough to like the only thing that seems to be very constant is the 16S. And so if all these different strains have different functions, then it's hard to kind of keep that locked down to the phenotype that you want to put in there.
Okay, thank you.
Quick question, quick answer.
So this is Ami Bhatt from Stanford. That was a great talk, and I think you've highlighted very nicely the importance of horizontal gene transfer on biological function of microbes within the microbiome. My question is whether or not you've looked for evidence of horizontal gene transfer in your FMT samples, so genes that are actually traveling from the donor to the recipient microbiome.
Yeah, so not yet. But now that we have all the donor strains, so we have a study going on right now that used the donor that we characterized really well with like the 2 years and all the isolates from donor 44. That donor we've used in a fecal transplant study for an inflammatory bowel disease, and we'll get samples back, and so we will go back and then culture those and see what came up. And so pretty excited to get our hands on those samples, but yeah, we don't have a story yet.
Great, we'll stay tuned.
Okay. Thank you so much, Eric. We'll get started on the next session.
Is somebody else using my computer? I think Peter is. He's using mine.
I think so.
Okay, good morning, everyone. My name is Padma Marwada. I'm a program director in NIDDK and also a member of Trans-NIH Microbiome Working Group. On behalf of our working group, I would like to thank all the attendees and the speakers who have readily agreed to accept our invitation and also present your work and participate in this 3-day event, which is not very easy considering your busy lives. And I would also like to take a moment, and I would like to actually give them a round of applause at this point. So I would also like to take a moment to recognize Lita Proctor. and applaud her for her effort to keep us, on task, and also spearheading the whole effort, and completely, keeping this momentum going for more than a year and a half. So I really would like to give her a round of applause as well. So the next two concluding scientific sessions actually focus further on translational aspects of microbiome and further explore and how the knowledge that we gained so far can be used for improving human health and also for disease prevention and treatment. And we have 4 speakers in this session: Peter Turnbaugh of UCSF, Scott Bultman of UNC, Maria Gloria Dominguez Bello of NYU, and Robert Jenk of MD Anderson Cancer Center. I request all the speakers to come up onto the podium just to save time. Of course, Peter is here, but others so each speaker is going to get 15 minutes, and I hope everyone sticks to their time slot in the interest of keeping the sessions going. And we will take all the questions at the end of the session. All the speakers have completed their talks. And also, I want our speakers to indicate if it is okay for, taking photos of the slides presentations. And I also request all the audience to respect and honor their requests. And Peter will give the first presentation toward metagenomic basis of therapeutics. Peter?
All right, so it's really a pleasure to be here. Thank you all for sticking into the bitter end today. And it's been a two days of really inspiring and exciting research. It's been very intimidating to go after Dr. Alm. So I'm going to tell you about the topic that we've really been focused on for the last few years in our lab, which is the important interactions between the microbes that live within our gastrointestinal tract and xenobiotics, including drugs, dietary compounds, and other environmental contaminants. And so I just want to start by pointing out that we're not the first group to think about this. Unfortunately, we've been scooped by almost a century. Back in 1939, Gerhard Domach was awarded the Nobel Prize for his really remarkable discovery that these azo dyes, which are commonly used in textiles and tattoo ink, have antibacterial properties. And this led to the development of Prontosil, shown here, which is the first drug in the sulfonamide class. And what Gerhard actually didn't realize at the time, despite the fact that he got the prize for this, is that Prontosil is actually not the active compound that kills bacteria. It turns out researchers at the Pasteur discovered that Prontosil gets cleaved into these 2 downstream metabolites, one of which, sulfanilamide, is actually the active compound that has the antibacterial effect. Even more interestingly, there's no enzyme in the human genome that performs this biotransformation. It's only performed by bacteria. And so you either need the pathogen itself or the microbiome to activate this prodrug, or inert compound, forming the antibiotic. And so I think this is exciting in the sense that it sort of led to the dawn of antibiotics, but also really was an early lesson that the drugs that we use in patients are really importantly influenced by the microbiome. If we fast forward to today, we now know that at least 50 different drugs are metabolized by gut bacteria, as well as bacteria in other body sites. And this list continues to grow, although at a relatively slow pace, year by year. But one of the things that's really, I think, interesting about this is that these drugs are very distinct from each other in their structure, as you'll see in these few examples on the slide. But they're also used for many different diseases. And so this is not just restricted to antibiotics like we talked about for Prontosil. It includes really key compounds that are used for the treatment of cancer, Parkinson's, rheumatoid arthritis, and others. And many of these drugs are really the mainstays of chemotherapy for these indications, even today. And so, unfortunately, despite the fact that many of these biotransformations have been known for decades, we still know very little about the enzymes that are responsible. And so that means that if we were to sequence a microbiome, it would be very difficult to predict whether or not it's capable of metabolizing any of these drugs. And so some of you may have heard the pioneering work in this area from this young scientist, Matt Redinbo. He at Poster 36 yesterday, and Matt really did amazing studies on irinotecan and the way in which the downstream metabolite SN38G is liberated by beta-glucuronases in the gut. Our lab has also worked on digoxin. We published the first paper a few years ago now, hopefully more to come soon. And since moving to UCSF 3 years ago, we've sort of expanded out to look at other medications. Others in the lab studying digoxin, as well as many of the other types of compounds we see on this slide. All of these people are finding, I think, really interesting things, and I hope to share the data with you guys soon in the near future. But I know that Padma is thinking, why is Peter talking about therapy? He was supposed to talk about prevention of disease. And so I want to switch for the rest of the talk today to an area related to xenobiotic metabolism that could have implications prior to the development of disease. And so first I just want to remind you that we are not only exposed to xenobiotics through drugs. Our diet is actually a rich source of small molecules. We all know that diet is a major risk factor for disease. But most of us sort of think about diet at these sort of macro levels, whether or not it's the actual food like a broccoli or a croissant. Whereas we should really think about our food in this way, which is that they are a complex mixture of small molecules that are not only the macronutrients that everybody talks about all the time, like carbohydrates and protein and lipids, but also a rich array of compounds that enter our body on a daily basis and are then modified by bacteria in ways that change their bioactivities. And so our lab is really interested in applying the lessons that we learned from drug metabolism to better understand the pathways through which gut bacteria biotransform dietary small molecules. And so I'm going to focus on just one class of compounds. This is work done by Elizabeth Bess, who's a chemist that joined our lab a couple years ago. And she, like myself, is obsessed with coffee and seedy breads. Interestingly, these are the two main sources of these polyphenols called lignans. And they're also found in cruciferous vegetables, as well as really ubiquitous throughout our diet. So all of us consume lignans every day, but the concentration varies depending on our diet. And so the reason why we're interested in lignans as microbiologists is that the lignin itself is actually thought to be an inert compound delivered through our diet that has very little effect on the body. What happens is that after we consume lignans, as we heard about from Joanna yesterday, they get modified by a series of reactions catalyzed only by gut bacteria to form these downstream products called phytoestrogens, like enterolactone. And so the impact of these dietary compounds on disease depends on the degree to which the microbiome can activate them. The area in which lignans have been most well studied is breast cancer, although they've been implicated in many other diseases. And this comes from work by Joanna as well as others to show that in humans, if you look at people at the highest quartile of plasma enterolactone, there's a 45% reduction in breast cancer incidence. In rodents, you can actually administer the compound itself or bacteria that produce the compound, and work done by the Blaut lab in Germany. And that's sufficient to reduce tumor growth in animal models. Okay. And so how do we go from the dietary compound to the phytoestrogen? And so this is a really simple model of what life might look like in the gut if you're bacteria, looking for these compounds in the food. And so we consume the initial substrate, pinoresinol, for example. And our favorite gut bacteria, Agarthala lenta, an actinobacterium, widespread in humans, performs the first two reactions. The middle one's not shown here. So it's pinoresinol to laurisaresinol, and then to SACO. Seiko then gets passed off to Blautia, which does the next reaction. It goes back to Elenta or potentially other members of the actinobacterial phylum, and then that goes on to lactonifactor. And so it's a really, I think, intriguing example of a multi-step pathway that creates these active compounds from dietary substrates, but also requires the participation of many different bacteria for the full pathway to be performed. Okay. And then they go in and they have their effect. And so, unfortunately, for all of the steps in this pathway, we don't know the genes that are responsible. And so if you were to go to Metapsych or KEGG or any of these databases, you won't find any annotations for lignin metabolism by bacteria. And so, we would like to know how each of these bacteria performs their step. And so Elizabeth was able to take advantage of the fact that our lab's been collecting unique strains of Elenta from around the world. We have 27 distinct Elenta strains now that come from 3 different continents. And what she did is developed an HPLC-based assay where she can quantify pinoresinol, laurisaresinol, and Seiko. So because we're not adding the next bacteria in the pathway, it'll stop when we get to Seiko. And then she just quantified the conversion for each of these strains. And so what you can see is that 18 of the 27 make Seiko in green, and the other 9 leave the pinoresinol intact. We then sequenced the genomes of these 27 strains and annotated orthologous genes or gene families. And so each row is a gene family, and then we're just looking at the presence or absence, so the absent in light gray, blue, and then present in dark blue. And we've just organized them by the non-metabolizers on the left, the metabolizers in the middle. And then on the right here, this is a random forest analysis to try to find genes that predict metabolism. But I think you could probably find the signal without any sort of stats, because at the top you can see there's these 2 genes that perfectly match the pattern of metabolism. So they're found in all the reducing strains and missing in all the non-reducers. Both of these genes fall together in the Elenta genome, and actually there's only just one enzyme that we're calling benzyl ether reductase, or BEAR, and then a transcriptional regulator that's next to it that we call BEAR-R. And so this was sort of the first mystery, is that we went in looking for 2 steps performed by Elenta, and we only have 1 enzyme. And so because we're simple-minded people, we thought that the 1 enzyme just does both steps. And that kind of makes sense if you look at the symmetry in the reaction. Consistent with the role of BEAR in this reaction, if we add pinoresinol to Elenta culture grown in vitro, we see that there's a 1,000-fold upregulation of expression of BEAR. And what's even more striking is that if we look genome-wide, we find that BEAR is by far the most highly upregulated transcript. And so this is RNA-seq data from Elenta grown in the presence or absence of pinoresinol. Each dot is a transcript, and then they're just ordered by the expression level on the x-axis. And what you can see is that there's a lot of significant genes that go up and down in red, but there's this sort of one lonely dot at the top of the plot, and this is BEAR. So it's orders of magnitude more expressed than any other transcript. We also see upregulation of the regulator, although to a lesser extent. Okay. And so Elizabeth was then able to go on to clone BEAR into a bacterium that doesn't normally metabolize lignins. And so this is the model organism E. coli. This is an empty vector shown here. We don't see, so we only see pinoresinol. We don't see the downstream products. And then when we clone in BEAR, now we see 80% conversion to Seiko, and the remaining 20% is the intermediate compound. In addition to that, she was able to use structural modeling to predict residues that are in the active site of BEAR. And so she found these 2 tyrosines that we predicted, or she predicted, would be involved in docking the substrate, mutated them both to phenylalanine, and then repeated the assay. And now you can see that, again, the wild type is really efficient. The control doesn't do anything. And then the double mutant has impaired activity. So it's still capable of going all the way to Seiko, but to a lesser extent than the wild type enzyme. Okay. And so the challenge now is that, we want to know all the steps in this pathway, but, we have a lot of tools for Elenta, but we don't really have any for Blaudia or lactone factor. And actually, I'm not going to go into it, but we don't actually think this step is performed by Elenta. It's potentially performed by other related bacteria. And so how do we now identify the steps in this pathway without needing to develop a strain collection for each of these bacteria? And so sort of inspired by the RNA-seq results for pinoresinol as well as other compounds we've looked at, we've been doing just incubations of each of these bacteria with the substrate that they should metabolize, and then looking for upregulated transcripts as a quick way to look at candidate genes. And so what we find is that this is Seiko incubation with Blaudia. As you can see, it's a little bit messier than pinoresinol, but there's these two genes in the top right that are also in a single genomic loci. They represent a transporter and a methyltransferase, so this is a really good candidate for that activity. And then the same for lactone factor. Here now you can see there's these three genes that are really far removed from the cloud, and those are all also within a single genomic loci, and they represent two enzymes and a transporter. And so we're currently validating these in E. coli as well as the enzyme for the remaining fifth step. And so just to sort of wrap up this project, I want to explain to you why we think it's worth going to the trouble of identifying these novel genes that are involved in xenobiotic metabolism. So the first thing you could imagine is that once we succeed in identifying all the steps of this pathway, that provides a tool in which we could now engineer bacteria that perform the full pathway. And so what we're starting with is this complicated system where you have changes in diet, you have many different bacteria that are involved, changes in the concentration of these compounds, and then ultimately the phenotype people care about, which is reduction in breast cancer or other diseases. And so we'd like to merge all of these into a single organism that performs all the steps in the pathway and in which we could regulate the activity of each step. We also now have the ability to perform germ-free mouse experiments in models of breast cancer. And so together with Jay Debneth at UCSF, we've developed nodobiotic mouse models of breast cancer, and we hope to colonize them with these controlled strains representing different steps in the lignin pathway, and see whether or not that results in a change in tumor burden. Okay. And so I just want to end with a topic that I think has come up throughout many talks throughout the last couple of days, and that's I think in order to understand what's going on in the microbiome and in general in host microbial interactions, it's going to require a lot of us that are biologists and metagenomicists and comfortable with microbes to start thinking more deeply about the chemistry that's performed by these microbial communities. And so just like Walter White was inspired by his battles with cancer to become a chemist, we as microbiome researchers need to learn and go back to chemistry, biochemistry, structural biology, and enzymology in order to better understand how these microbial communities function at a molecular level. And so if you're interested in sort of meeting chemists or hanging out with chemists, maybe becoming one, we're, together with Emily Balskos and Dennis Wolin, we're organizing a Keystone Conference for the next session sometime in 2018 to 2019, so stay tuned. So I just want to acknowledge all our wonderful members of the lab. Elizabeth Bess is in the middle of this cluster here. You can see she's the postdoc leading the lignin project. I also want to acknowledge David Gutenberg, who's here at the meeting somewhere, hopefully he's still around, who was the first person to join my lab when I was a fellow in Boston, and it really had a transformative effect on getting us started and all of the early work we did when we were on the East Coast. So thanks a lot, and thanks for having me.
Thanks, Peter. So the next speaker is Scott Bultman, and he's going to talk on gut microbial metabolites and cancer prevention.
Okay, so thanks to Lita and the other organizers for this opportunity. I'm having a great time. And so our lab is interested in the idea that dietary factors and digestive components get metabolized by gut microbiota into metabolites that influence the risk of various diseases, including cancer. And so So this can impinge on a number of different cancer mechanisms. And so, for example, certain things like red meats and bile acids can get converted to putative oncometabolites, whereas other things like plant-based fibers and polyphenols can get converted over to putative tumor-suppressive metabolites. Now, these metabolites can have direct effects on the intestinal epithelial cells. They can also affect the immune cells to modulate the immune response and inflammation. So, should go back one. So we're especially interested in this guy here, butyrate, and it's a short-chain fatty acid. And if you look at its structure, you can really see for yourself that it is a short chain with only 4 carbons. And I do know that there's a handful of short-chain fatty acid and butyrate aficionados in the audience, but for those of you that aren't, the story really begins up here with dietary fiber. And so this is the complex carbohydrate that's not digested or absorbed in the upper GI tract. And it reaches the colon, and here there are specific clades of bacteria that ferment it into short-chain fatty acids. And so these are the 3 most abundant. They're each present in the lumen at millimolar levels. And what makes butyrate interesting to me is that it's primarily utilized in the colonic epithelial cells themselves. And so it gets transported in, and it can go to 2 different subcellular compartments. First, it can go to the mitochondria, where it's important for energetics. So unlike most cell types in our body that use glucose as their primary energy source, colonocytes are different. They get like 60 to 70% of their energy from butyrate. I think the idea is that there's a lot of it, and as a fatty acid, it's oxidized readily. Now, secondly, butyrate can also go to the nucleus, where it's important for epigenetics. It's a naturally occurring histone deacetylase inhibitor, or HDAC inhibitor. In fact, arguably the first one ever characterized. So there's a couple lines of evidence that implicate butyrate in cancer prevention. First is if you add butyrate to tumor-derived cell lines, including colorectal cancer cells, it decreases proliferation and increases apoptosis and or differentiation. So this is a great result, but I think we're all aware of the caveats of tumor-derived cell lines that I'm not going to get into. So it's good that there's a second line of evidence, and it comes from 16S sequencing studies. I'm showing a few studies here, and these are all working with human fecal samples. And the good news is that they all reach the same conclusion, and that is for butyrate producers, their number or abundance is actually diminished in the colorectal cancer cases compared to controls. And so this is also a really good result, but there's a limitation here too, and this has been kind of a recurring theme at this meeting. It's really difficult or impossible to know whether a particular change in the microbiome is a cause or a consequence of disease, and that limitation holds here for sure. So we've taken a slightly different approach to further interrogate butyrate function, and what we're doing here is we're using gnotobiotic mouse models. And so I'm depicting 2 isolators here. I'll start on the left. So these mice were germ-free, and now we've colonized them with 4 commensal bacteria referred to as the altered Schlatter flora. And so these are commensals that occupy different GI niches, and they restore some degree of normalcy to the gut, which is abnormal in a germ-free state. But it does this in a very controlled manner. And then we provide the mice with control or high fiber diets that are calorically matched and virtually identical. And the high fiber diet in this case is 6% fructooligosaccharides and inulin, and we chose that because it's fermented really efficiently into butyrate. And then we induce tumorigenesis. We're using the procarcinogen AOM, or azoxymethane, and this only induces colorectal tumors, and there are a lot of similarities with human sporadic colorectal cancer. For example, it mutates either APC or beta-catenin, leading to deregulated Wnt signaling. Over here, it's the same scheme, except for these mice have been colonized with the 4 ASF bugs, plus this guy here, B. fibrosolvens. This is a butyrate producer. Other than that, same scheme, control diet, high fiber diet, induced tumorigenesis. And so the idea now is control, 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 experimental, because it's the combination of the high fiber diet with the butyrate producing bacteria that yields high levels of butyrate in the lumen of the gut in the first place. And we know this based on LC mass spec measurements. So the hypothesis then is that as a result, the experimentals will have a diminished tumor burden. And so this is in fact the case based on a number of experiments. So when we look at this graph, first, the high fiber diet on its own, in the absence of the butyrate producing bacteria, does not have a protective effect. And similarly, the butyrate producer on its own, when it's matched with a low fiber diet, also does not have a protective effect. Instead, it's the combination of the high fiber diet with the butyrate producing bacteria in these experimental animals that gives rise to significantly fewer tumors. In addition, these tumors are smaller, and when we do H&E based histopathology, they're less progressed. So we needed to do another experiment to really show that butyrate is in fact a causal factor. And so what I'm showing here on the left is kind of re-showing what I just showed you, the 3 control groups in the experimental with the wild type butyrate producer and the lower tumor burden. Now over here in this isolator, it was the same experiment, but we're working with a mutant strain of B. fibrosolvens. So it's the same bacteria, different strain, 0.8 kb deletion in the middle of the butyryl-CoA synthesis operon. And these bugs have decreased, diminished butyrate production. And so when we match this with the high fiber diet, there is a tumor suppressive effect, but it's significantly attenuated compared to the wild type butyrate producer. So in other words, the tumor suppressive effect is commensurate with the butyrate producing capacity of the bacteria. And so we did one other important control experiment, and that's shown on the right side here. So these mice only got the ASF bacteria, no butyrate producer, and instead of being given the high fiber diet, they were given the control diet, but fortified with butyrate. And this had just as strong or stronger of a tumor suppressive effect. So this shows that we can recapitulate the high fiber diet by wild type butyrate producer effect by supplying the mice with exogenous butyrate. So we've done a lot more to kind of develop a working model. There's aspects of it that I'm not going to be able to get into any kind of detail today. But what it is then is fiber, bacterial fermentation into butyrate. Butyrate is important for just normal colonic homeostasis, in large part because it's metabolized in the mitochondria. It has more to do with energetics. And then it is important for tumor suppression. Now in the tumor cell, these cells, due to the Warburg effect, switch their metabolism such that they are more reliant on glucose metabolism. The butyrate transporters are still expressed, butyrate's still transported in, but now because it's not metabolized to the same extent in the mitochondria, it actually accumulates to higher levels, goes to the nucleus, can act as an HDAC inhibitor to epigenetically regulate gene expression that do these things. There's a number of things that still need to be done with this model to flesh it out further. One is to get at the idea of butyrate target genes. And the way that we're doing this is ChIP-seq to look at H3K9 and H3K27 acetylation. These marks are enriched at promoters and enhancers respectively, along with RNA-seq. And to give an example, we're seeing enrichment at a lot of cell cycle and pro-apoptotic genes, showing one trace here with FAS, and we're looking at H3K9 acetylation in the promoter, where you can see higher levels in the tumors from the experimental mice than the control mice. The other thing that we're really looking at to try to get an idea of where the specificity would come in is co-occupancy with 3 HDACs that are recurrently overexpressed in colorectal cancer. And so we're doing this now. The model that we have is that during tumorigenesis, the HDACs are upregulated and they're playing a role in driving cancer, and they are diminishing the expression of certain pro-apoptotic genes like FAS. And now if there's sufficient levels of butyrate, it can act as kind of a brake pedal on the driver to allow higher levels of expression to be reestablished. There's other things that have to be done as well. We need to investigate more thoroughly the role of butyrate in inflammation. We're doing this a couple different ways. We're looking at other dietary factors. And then the other important thing with mouse models, and this has come up earlier too, is human relevance. We have a little bit of data on that. So looking at nearly a dozen adenocarcinomas along with normal matched colon samples, we see, just like our mouse model, we see higher levels of butyrate accumulating in the tumors compared to the normal. And also, just like our mouse models, we see higher levels of global histone acetylation in the tumors compared to normal. So we think that this is encouraging. Now, shifting over to challenges, I have a couple. Challenge number one is the need to kind of do high throughput functional analysis of microbes. I think this problem is becoming even more acute because this notion that most of the microbiota are uncultivatable seems to be, the idea is diminished day by day. We're getting more and more microbial isolates, and we're going to need a way to interrogate their function. So I'm obviously a fan of gnotobiotic mouse models. But there are limitations, and I have a few of them listed here, including the fact that they're expensive and low throughput. And so there is a need to maybe develop cell culture models that will allow, that will be amenable to co-cultures with microbiota. I'm not talking about Keiko-2 cells. I'm going more in the direction of mini-guts and organoid technology. So this is a huge advantage, but this also has limitations. So this is the typical state of the technology, and they do grow as spheroid structures. And so the pseudolumen is enclosed in the middle here, and it's very difficult to control the conditions, and this is where the bacteria would be. So the bacteria actually need to be physically micro-injected inside. And so this isn't ideal. There's room for new approaches, and my colleagues and I are working on this. And so what we have is we have a scaffold, and it's shaped the same size and dimension of a human crypt. And it's coated with hydrogel, shown in pink here, and then seeded with human LGR5 intestinal stem cells. And one of the advantages of having this in an open format where it's not enclosed as a spheroid is that we can apply chemical gradients and growth factor gradients, including Wnt. And this allows the crypt to be polarized such that the stem and the progenitor cells that are proliferative, which are shown in green, are at the base of the crypt, and the postmitotic differentiated cells shown in blue are near the lumen. And to really show you this, here is an in vivo human crypt, and here's one of our in vitro crypts on the device. And so if we look at the green marker, we have the progenitors and the stem cells at the base, and if we look at the red marker, we have the differentiated cells near the lumen. We can co-culture these with microbiota. What we're really doing first is trying to deliver a sharp oxygen gradient, kind of depicted here. And we haven't done this yet, it's still ongoing, but the hope is to keep the crypt in a normoxic state that will keep the colonic epithelial cells happy, but it would have to be a very sharp gradient, of course, and keep the luminal compartment anaerobic. And so this will allow for co-culture of obligate anaerobes, which would get us at host microbe interactions that are really physiologically relevant. Another challenge is kind of moving from identifying metabolites to really understanding their function. So the first phases of the human microbiome project make sense. Perfectly appropriate to take discovery-driven multiple omics approaches, and it's yielded a lot of information. I like this approach, obviously, because it's unbiased. It covers a lot of ground, kind of like plowing, to use a farming analogy, but the limitation is it only scratches the surface. And so hopefully at some point we're going to be at a point where we have a list of interesting candidates, and the challenge is how that's vetted and how we move into the next phase, which is more hypothesis-driven, where we look at specific candidates, and to stick with a farming analogy, where we drill down and probe function more. But this takes a lot of time, and it's something that we'll need to discuss. One idea on that is that there could be some challenges on this. A cautionary note comes from butyrate. It's a metabolite that we know relatively a lot about, it's pleiotropic, and probably a lot of these metabolites are going to be pleiotropic. So, and they act by multiple mechanisms. So butyrate's an energy source, it's an epigenetic factor, it's a ligand for G protein coupled receptors. It can act on cell autonomously on the cancer cell itself, but it can also act non-cell autonomously via the immune system. This can either be indirect through barrier function or direct by inducing the regulation of Treg cells. And so trying to figure out the relative importance of all of these different mechanisms is not trivial. And then my last thing, challenge number 3, trying to improve, move things into the probiotic and prebiotic realm in an accelerated manner. This can take a lot of forms. One of the things would be to integrate microbiome studies a little bit better with epidemiology and GWAS, or exome sequencing. And so I'm over, so just acknowledge folks in my lab. Nancy's the biomedical engineer, and Scott's the stem cell person that really is largely responsible for the organoid project. And with that, I'll stop. Thanks.
So the next speaker is Maria Gloria Dominguez Billo from NYU. She will be talking on microbiomes and phenotypes, impact, restoration, and transfer.
Thank you very much, Padma, and thank you, Lida, and the organizers of this wonderful symposium. Humans started an urbanization process that has lasted and is continuing through the last few thousand years. And we know that the world is becoming more urban every time, and with urbanization, we have the well-known immune-associated disorders. So there are two hypotheses. We don't know why this is happening, and there are two hypotheses that deal with microbial etiology. One is the hygiene hypothesis, which deals with the effect of exposure to environmental bacteria. And the other is the disappearing microbiota hypothesis, which postulates that it is human bacteria from the microbiome. The fact is that as mammals, humans start life with a heavy exposure to human bacteria, maternal. We are all born, not if we are born naturally, already seeded, already inoculated by the time of birth, picking up the colonization bugs from our mother's vaginas. And then the babies will not eat but a liquid produced by the mothers, which is milk. And that will select for some of the vaginal perineal bacteria that were picked at birth. And then mom is still there until the baby really develops enough sensorial brain locomotion to be able to explore the environment. And there comes a very deep exploration of the environment after that. And in urban societies, because we have technologies that have been very efficient for extending lifespan, but we perturb and abuse practices that really impair transmission and colonization of both microbes. On the human side, we have C-sections and antibiotics, formula feeding, and we use technology to keep the baby far from the mothers. to transport them or monitor them in their own very clean rooms where they are raised. And the first big impact at birth, during labor at birth, is C-section. C-section is a composite because it requires also antibiotics. So it's bypassing the birth canal and the perineal area and antibiotics. C-section has been related with the modern diseases as have early antibiotics. It's the same diseases that are increased in risk. We showed earlier, in collaboration with Rob Knight, how C-section impacts the neonatal microbiota. If we compare with maternal sites, oral, vaginal, or skin, the babies will cluster, any body site of a neonate within the first 24 hours will cluster with the vaginas if the baby were delivered vaginally, or with skin if they were by C-section. This is a metagenomic view of the vaginal microbiota. This is work by my graduate student Keith Martinez, and Amanda Birmingham help us do the analysis. So we can see it's mostly Firmicutes, Lactobacillus, and Proteobacteria with a little Bacteroidetes and Actinobacteria. This is the vaginal inoculum that is massively transferred to babies. Now let's look how the baby gut selects after 2 days of getting this inocula. And what we see is, at the expense of the Firmicutes, Bacteroides and Actinobacteria really expand. How does a C-section born baby look at 2 days? They really lack the Bacteroides and Actinobacteria component. This is at day 2, and these are pools because we are using these to humanize mice. But we know that there are longer term effects. The communities between the babies converge, but there are populations that never do. C-section babies sustain a low Bacteroides development in their gut. There are alpha diversity differences that emerge after the 8th month. So we see convergence in alpha diversity, but eventually those babies around 1 to 2 years of age, they will have lower alpha diversity than the vaginally born. And there are sustained beta diversity differences. So C-section is a big impact, no wonder, it's expected. But how about smaller impacts? For example, going to the hospital to give birth. So Joanne Kombelic, a PhD student, insisted, she's a midwife and was doing a PhD in nursing. She insisted she wanted to do home birth. And I warned her that, with the babies, we could handle the number of babies, we may not see any differences. So we took what we call green babies. Green babies are vaginally delivered without antibiotics and all exclusively breastfed. She followed the neonates for the first month. And to our surprise, although no differences were seen in alpha diversity, they were in beta diversity, with hospital babies having significantly higher Enterobacteriaceae and home babies having higher Bifidobacterium, Bacteroides, and Streptococcus. It's a very subtle impact, if you want, stressor. We think what happens in the hospital is not the hospital air, although we don't know, but most likely hospital deliveries have always interventions, maybe labor induction or anesthesia or the episiotomy, and the fact that all babies in this country by protocol receive ointment antibiotics, for example, even if they are born by C-section. So interventions at birth do change the neonatal microbiota, and this may lead to developmental changes. So as ecologists, the first thing we thought is, okay, can we restore nature in some ways? And one way we can restore is exposing the newborns to microbes of the vaginal tract. So we did a pilot study, and we are now analyzing the bigger study data. in which healthy mothers with healthy pregnancies, negative to infections, that had to have a scheduled C-section due to malposition presentation or repeated C-sections, where they were inserted a gauze in the vaginal canal 1 hour before birth, and then we used that gauze to immediately after they cut the umbilicus, as soon as we could, expose the baby in all body sites, starting with the mouth. And we showed in that pilot paper publication how the bacterial sources reflected in the different, these are babies born vaginally or by C-section, three body sites, and these are the sources. So the first thing to note is how rapidly, within 1 week, the anal site becomes typical, the oral site or the skin. And in the vaginally born babies, we have the purple source of vaginal bugs, not in the C-section throughout the first month. And exposing them to the gauze restore at least partially that signal that was lacking in the C-section. And we can see what populations, and this is 16S based here, anal Bacteroides and Lactobacillus were restored, skin Lactobacillus S24/7. So if you expose a baby that is taken out through an orifice artificially to the world and expose that baby to vaginal bacteria, they will pick up vaginal bacteria. So that's fine, that's microbial restoration, but so what? And many mothers ask that. We don't know yet if it hasn't been proved that there is direct disease causation by the C-section or whether this microbial restoration protects. So here are the gaps. To test whether microbial restoration protects against the diseases, we need randomized clinical trials to assess different diseases that require to follow babies for 5 years, and it's expensive with a lot of babies. Also, we need to know which are the taxa that are normalizing if it works. So we need animal models and metagenomics, strain discrimination approaches. It's expensive. We also lack the appropriate technical culture capability. So one question we can ask is, given the multiple and commonly used perinatal stressors that follow, in addition to the ones I mentioned, there is processed food afterwards. Can we ask the question, are we impacted? And that we can compare ourselves with tribal peoples who have not been exposed to medical technology. We did that initially in collaboration with Jeff Gordon and later with Rob Knight and Jose Clemente. We first showed that the US has the lowest diversity gut microbiome in relation to Africans from Malawi or Guahibo. These were semi-transculturated or transculturated rural people. And then we published the highest diversity microbiome so far from uncontacted Yanomami Amerindians. And we could follow the OTUs, and the prevalence abundance curves show that in relation to the green Amerindians towards the yellow US, we lose abundance and we decrease prevalence. In a recent study that will be published very soon in Science on the Hadza from Tanzania, we show how the fecal microbiota moves along the PCO1 in the PCOA, and the trajectory of the microbiotas really orders in function of lifestyle from jungle to rural to urban, confirming that urbanization is indeed associated with changes in the gut microbiota and losing diversity. So the HMP microbiota is an impacted microbiota. Another gap then is we need to study the unimpacted microbiota and understand what is the lost microbes, what are the lost microbes, and what are the functions that we are losing. In collaboration with Peter Dorestein, we did urine metabolites across urbanization in Amerindians, from red low urban levels to higher rural levels in orange, and a control mestizo. We see the gradient of urbanization in the urine metabolites. So people do change. In another gradient of urbanization, we decided to study homes to address the environmental differences. And we could tell, in a published paper with my student Gina Ruiz, we could tell apart the walls of the houses by the urban site. And so with urbanization, we increase human bacteria in the houses and reduce soil. We did metabolomics in collaboration also with Peter. And again, we see in the floors and walls a gradient of urbanization in the chemical similarities between the surfaces of homes and our skin. And the reason were mostly our practices. So in Checherta, in the jungle town, we see a lot of breakdown of chlorophyll and other natural products. In the city, we see sweeteners, analgesia, analgesics, antifungal, medicine for high blood pressure, epilepsy, reflecting the diseases being treated. So urbanization is associated with reduced soil and plant exposure and increased exposure to synthetic chemicals. So my last two gaps, gap 4, we need more evolutionary approaches to study the microbiome, the impacts and consequences. We need to make of the microbiome an anthropological discipline. Finally, the last gap is how do we come with restoration? Especially which taxa, and what about if it has been lost? So to protect future generations of these diseases, the same way we are conserving, we have a list of endangered species of animals, and there is a global seed vault in the permafrost in Norway. We should definitely be constructing for the future a human microbiome vault. We don't know the functions yet, we don't know the utility, but hopefully future generations, science will have these resources to apply for human health. And with that, I acknowledge the funding, the people in my lab at NYU, my students at UPR, and my very valuable collaborations. And thank you for your attention.
The final presentation for this session is by Dr. Robert Jenk. He'll be talking on allogenic hematopoietic stem cell transplantation, clinical outcomes, intestinal bacteria, and potential mechanisms.
Hi, good morning. My name is Rob Jenk. I work at MD Anderson Cancer Center. I moved there recently from Memorial Sloan Kettering, where I was doing my training and worked as junior faculty for many years. The title of my talk is Allogeneic Bone Marrow Transplant, Clinical Outcomes, Intestinal Bacteria, and Potential Mechanisms. I should say I'm a clinician. I am not a microbiologist, although I have tried to pick up some stuff over the years, I guess. I wanted to start with my unmet need, and I think this ties into what other people have said during this meeting. There's a lot of very interesting and fascinating associations that we've seen. There's very, very good preclinical modeling that's been done to address the question of causality, but one unmet need, I think, is clinical trials, well done, randomized clinical trials to help spur efforts from biotech to decide which diseases to focus their efforts on. And a lot of these studies cannot be done without either federal funds or philanthropy funds, because these are agents that can't be patented, right? These are antibiotics or prebiotics or probiotics or fecal transplants. You can't patent these things, at least not very easily. I found this picture last night of Dr. Ben Eisman. He's a surgeon who was at Denver in Colorado for many years. And in 1958, he reported in the Journal of Surgery 4 patients with pseudomembranous colitis. This preceded the identification of C. diff. And they reported that they treated these patients with fecal enemas, and all 4 patients responded within days and were discharged from the hospital. So this is 1958, and then I should mention fecal transplant was championed by Tom Borody, a gastroenterologist in Australia, for many years. But it wasn't until 55 years later when a randomized trial in the New England Journal done by some Dutch investigators, not Americans, and this really led to the idea that the microbiome could be a therapy. So that's my soapbox gap to highlight. So a little bit about bone marrow transplant. This was originally developed as a means of rescuing patients after high-dose cytotoxic therapy. The concept was if you can give patients bigger and more toxic doses of chemotherapy or radiation, can you cure more patients? And it turns out you can. Eventually you start to hit a wall of toxicity, and one of those walls was marrow failure. And some clever scientists were able to figure out that you could rescue patients by taking out their marrow, giving them the chemo or the radiation, and then reintroducing the marrow, and suddenly that wall at least is eliminated, and you can give higher doses. Later on, some other clinicians were trying to use bone marrow grafts that were cancer-free, so they went on to use a bone marrow from a different individual, an allogeneic bone marrow transplant. This led to, for particular cancers, higher cure rates, but at the same time, what the investigators found was that there was this dramatic inflammatory immune response that was mediated by the donor bone marrow, the donor immune cells. And that syndrome was called graft versus host disease. And that continues to be a problem. This is CIBMTR registry data showing how we fail our allogeneic bone marrow transplant recipient patients. The biggest slice of the pie is disease relapse, so that's their malignancy coming back. But the second biggest piece of the pie is GVHD. This is what's generally known about the pathophysiology of GVHD. You start out with the patient who undergoes host conditioning, so they undergo chemotherapy, radiation, they get the cells infused, and early on in the process, bacterial products, so primarily bacteria in our intestinal tracts, they release all kinds of inflammatory mediators, and these are thought to activate host antigen presenting cells, which in turn activate donor T cells that are graft derived, and then this leads to a whole inflammatory cascade. Eventually this leads to inflammation of the classic afflicted target organs: skin, GI tract, liver, hematopoietic system. And you can also have a beneficial immune effect against the tumor as well. So I've highlighted in the pathophysiology the bacterial products. And so these early bone marrow transplanters, they were able to show actually in a very early time period that the microbiome is influencing this disease process. And so they did this in 2 ways. One was with germ-free mice in the 1970s. They showed that mice that were irradiated and transplanted in the germ-free setting had minimal graft versus host disease. And then there was another group that showed that you could get a similar benefit by administering gut decontaminating antibiotics. So this led to clinical trials of gut decontamination in patients, sometimes with positive results, other times with mixed results. And as a result, the practice sort of fell out of favor, and the field doesn't really know exactly what to do in terms of the microbiome. So this is our sort of scheme. We've been collecting stool samples from patients undergoing bone marrow transplantation, sequencing them by 16S, and then trying to associate with various clinical outcomes that we're interested in, bloodstream infection, survival, GVHD, relapse. We started in 2009. This is actually a wonderful collaboration between two physician scientists at Sloan Kettering. My former mentor, Marcel Vandenbrink, is the bone marrow transplanter, physician scientist, and Eric Paymer, who's in the audience and speaking later, was the infectious disease physician scientist. And the two head honchos got together and set up this collaboration. And as of when I left Sloan Kettering, we had collected 3,000 stool specimens from over 800 patients, and that's been ongoing since, and I'm sure is much, much higher now. So this is what the data looks like. Each sample here is a stool sample collected from a single patient during his transplant hospitalization, and with the colors, we're showing you the composition. So you can see that the patient started out pretty diverse with lots of Clostridialis, and was actually able to maintain that diversity throughout much of the hospitalization, despite some antibiotics that he received, vancomycin IV and aztreonam. And then around day 30, something happens, and suddenly there's a bloom of green bacteria, which is Enterococcus. And it turns out that was actually VRE, which we heard about yesterday. And that coincided with a diagnosis of C. diff infection and treatment with metronidazole. So I thought this was interesting. It was interesting that certain antibiotics impact on the microbiome more than others, and that there's heterogeneity in these patients in terms of how well they're able to maintain their baseline, generally healthy microbiome composition. So we asked the question, is there any association of certain bacterial genera with dying from GVHD? And we asked this question in a cohort of 64 patients. Here are the results using Curtis's LEFC tool. And I've depicted the p-value on the y-axis, and it's inverted on the log scale. So a better p-value is higher up, and hits to the left are associated with less GVHD, hits to the right are associated with more GVHD. So our top hit was a genus called Blaudia, and it was associated with less GVHD. So we stratified the patients based on their Blaudia abundance into two halves. The patients with more Blaudia are in blue, and they did very well. They had a very low incidence of mortality from GVHD. The patients that had less Blaudia did very poorly. We were lucky to have a second cohort of patients to try to validate or reproduce this finding, and it actually reproduced very nicely. And then here's the combined curves with overall survival stratified by Blaudia abundance. So we thought this was interesting. There were other groups in the field that were also asking similar questions. This is a study from Ernst Haller's group in Regensburg, and they were actually able to identify a urinary metabolite of microbiome health. Urinary indoxyl sulfate is, I guess you start with oral tryptophan, which is then metabolized by your gut microbiome into indole. And he actually was able to show an association with Clostridia potentially mediating a lot of that fermentation. So the indole gets absorbed, gets metabolized in the liver, and then comes out in the urine. And so they're measuring these urinary metabolite levels, which is, I think, very interesting because this is potentially a way, a quick measure of microbiome health that doesn't rely on 16S sequencing, for example. So they were able to show that this was prognostic for treatment, transplant-related mortality. And then there's a friend of mine, Andy Coe at UT Southwestern. He's a pediatric transplanter, and his group was able to find a very similar finding in that patient population. So then we went on to ask, well, it looks like antibiotics are a major mediator of this heterogeneity that we see in this patient population. So what are the effects of antibiotics on GVHD? And there's a nice insight here in that if you go back in time and look at the patient records, you actually don't necessarily need stool samples. You can just look at their antibiotic exposures based on their electronic charts and try to connect those with the outcomes. So that's what we tried to do. We looked at 850 odd patients at Sloan Kettering that underwent allotransplant over a 23-year period. So here's the incidence of GVHD mortality in this population, about 15%, which is pretty typical. Here I'm showing you the most commonly administered antibiotics, and I've stratified the patients based on exposure, whether the clinicians decided to give the patient that antibiotic or not. The only antibiotics on this chart that had a significant p-value were piperacillin tazobactam and imipenem cilastatin. Both of these had a significant association with GVHD mortality. And incidentally, both of these are used to treat neutropenic fever, which occurs very commonly in these patients. So here are the incidence curves for pip/tazo on top and imipenem on the bottom. And for both of these antibiotics, the patients that received the antibiotic are in red, and they're the ones that did poorly. So this is interesting. It turns out there was another couple antibiotics that we use to treat patients with neutropenic fever, though, cefepime and aztreonam that I'm highlighting here. And these are given as second line to patients who have a history of a severe penicillin allergy. If they have a history of a rash, they tend to get cefepime. If they have anaphylaxis or hives or something like that, they tend to get aztreonam. And so these are the infectious disease folks in the room will also notice that these are more narrow spectrum antibiotics. They tend to spare the anaerobes, particularly aztreonam is very sparing of gram-positives in general. So it was sort of a natural experiment that occurred. And we looked at the incidence of GVHD mortality relative to exposures to these antibiotics, and there was no association. So we thought that was interesting. I mentioned earlier, I've since left Sloan Kettering and now at MD Anderson. And so we did a similar study there and looked at almost 300 patients, looked at their antibiotic exposures, and found that similar results were shown. Meropenem patients had increased GVHD, while cefepime patients did not. So then we went on to use a mouse model of GVHD and administered these same antibiotics, and this actually recapitulated the findings. Imipenem was associated with aggravated GVHD in the mouse model, as aztreonam was less severe. The effects were localized primarily to the large intestine, and when we looked at the microbiome, we found that there was actually a bloom of Akkermansia, which is something that we've been hearing about in this meeting multiple times. It's mucin loving, and so we looked at the mucus layer using this Carnoy's method, and we found that imipenem treated mice had lost their mucus layer. And there is actually bacteria that have translocated into the lamina propria. This led to impaired barrier function. And so then we asked, well, what if we can try to get rid of all the bacteria, including the Akkermansia? So we added oral ampicillin to the drinking water, and we found that indeed, on the bottom right here, that those mice, despite imipenem treatment, were spared from the thinning of the mucus layer. And this led to, this is quantified here, and this actually led to reduced GVHD severity scores. So this is sort of our model, our framework. We think that if you can avoid antibiotics and you need a bone marrow transplant, you're probably in good shape. If you receive high disruption antibiotics like pip/tazo or imipenem, then this leads to a high GVHD risk state. And then in terms of what can you do, well, one option would be to push people all the way to the other end of the spectrum where they're basically gut decontaminated. And then alternatively, you can try to push them back to the sort of original healthy state of the microbiome. So I want to acknowledge my former mentor, Marcel Vandenbrink, our very close collaborator, Eric Paymer, their laboratory members that did a lot of this work, my clinicians, clinical colleagues, the nursing staff for collecting all these thousands of stool samples, and collaborators, as well as my funding sources. Thanks.
Questions for the speakers? Please identify yourself before you ask the question.
I'm Lakshmi Goel from Cell Host and Microbe. My question is for Dr. Domingo Bello. C-sections were quite common, I think, a few decades ago. So is there any epidemiological data that associates C-sections with health or disease outcomes? And along those lines, could you then take your cohort and follow them, the restored cohort, and follow them over time for those disease and health outcomes?
That's a very good question. I think there is a compelling epidemiological association in big cohorts showing that C-section increases the risk of asthma, celiac disease, and obesity. The direct causation and the mechanism hasn't been shown. Our cohort is too small. We are following those kids, but we have only 84 kids. It's very small. And so to assess effect on health, we need about 1,000 children. We don't have the money to do that.
I'm Janet Jansen from the Pacific Northwest National Laboratory, and my question is also for you, Maria. I was very intrigued by your idea for a human microbiome seed vault. And I was wondering, for the samples that are extremely valuable from the Amerindians that you have, is there any attempt to cultivate isolates from those samples?
That's a very important question. We have the specimens in my lab. We have backups in Venezuela. So you can imagine how risky they are at this moment. And that's part of why it really led me to think that we should save that for the future. We have done some cultivation from fecal. We have oral specimens and skin. Those collections are subject to some restrictions, like we cannot patent anything on them. Everything has to be cultured. But I would like to have collaborators to improve the success in culturing and also do comparisons of evolution of these microbes on those people who were isolated for 24,000 years and the rest of the Asians or Western populations. That's something to be done. Very little has been done, basically because of my limitations.
Thanks. These were great talks, and I have two questions, but I think there are two short answers that could be gotten. First, the question is to Peter, and I like the idea of engineering microbes to do this metabolism to get the active product, but the more practical solution might be just administering the active ingredient, and I think that you can solve a lot of delivery issues as well as colonization issues with the superbug. I was wondering what your thoughts on that were.
Thank you for the question. No, that's a great point. I guess what I didn't really get into very much for phytoestrogens is that in breast cancer, as far as I'm aware, most of the epidemiological data supports a preventative effect of the phytoestrogen. So that is something about sort of long-term production of phytoestrogen prevents the development of cancer. There's a lot less data on treatment of cancer with phytoestrogens. Even in animal models, typically the experiments are done where the phytoestrogen is given prior to or at the same time of initiation of cancer. And so, it's not super clear whether or not that'll ultimately be effective. I think there's also a potential advantage in being able to deliver the compound at the same location where it's naturally produced. No one has really done, as far as I'm aware, careful studies of what happens if phytoestrogen is produced in the small intestine versus large intestine, and whether or not that would have a difference in outcome. But really interesting.
Thanks. So my second question is to Dr. Jenk, and I like the idea of this acromantia and then erosion of the barrier, but is it possible that the more narrow spectrum antibiotics also preserve microbial groups that might be immunotolerizing?
I agree with that completely. We actually did look in that study to see if there were increased regulatory T cells in the setting of the narrow spectrum antibiotic. We also tried to measure butyrate levels from the stool, and there was actually no difference, and that's why we had to sort of go hunting for an alternative mechanism, and we stumbled upon this Akkermansia mucin finding.
Okay, thanks.
Hi, my question is for Maria. Thank you. Those were all great talks. I wanted to ask about the inoculation of the baby. So the data on C-sections done if the mother was in labor or had rupture of membranes actually shows that the outcomes are better for those infants with regards to allergies and development of diseases in the future. Is there an alternate way to inoculate these babies, perhaps by premature rupture? And also, are we discounting the effect of labor? Perhaps there are some hormones that affect the baby in utero, because even labor without rupture in babies delivered by C-section have better outcomes. So could that alter how they acquire the microbiome?
Those are very good questions, and I think there should be a lot more effort put in to answer them. We absolutely don't know. There has been very little effort put into optimizing labor, optimizing the natural way of giving birth, and studying why that is important. I don't have answers to your questions. Very good questions.
Hi, Brandon Eicher. Dr. Turnbull, your talk was fantastic, but I think it inspired a gap identification for me. We have millions of these compounds, and so I'm wondering how we might be able to take your work and speed it up a little bit and start doing more high throughput discovery in a similar manner, minus herds of undergraduates. How can we potentially put these on small chips or start to do some type of more metagenomic analysis so that we can get down to getting scores of these things identified and knowing that your gut bacteria are different than mine, and start to go towards more population level understandings of these things so we can better formulate diets and nutrition and drugs. Thank you.
Yeah, that's a great question. I think the solution is our lab needs approximately 500 RL1s, and then we'll just put one person on every compound. I think there's a huge need for speeding up the process. I think one thing that I should mention is even in these sort of reductionist approaches, what's proven true for human xenobiotic metabolism is that there's an incredibly limited set of enzymes that performs a vast diversity of reactions. And so, every new enzyme that was discovered in the human body that metabolizes a drug, taught us about many different drugs, dietary compounds, and endogenous compounds. What we don't know is whether or not that will also be the case for microbial xenobiotic metabolism. So, will the substrate scope of each of these enzymes be restricted to the compound that we're originally studying, or will it teach us about a broader array of compounds that are metabolized by bacteria? I can tell you, for the one enzyme that we've studied in the most depth that I didn't have time to get into, which is CGR, the cardiac glycoside reductase that metabolizes digoxin, work by Nitzan Koppel and Emily Valskis's lab has shown that interestingly that enzyme can metabolize many cardenolides, so that this is a broader family of toxic compounds that are in drugs as well as plants. But it does not seem to metabolize many other related glycosides. So, yeah.
I'm Mark Rhodes from Houston, Texas, and for more than 30 years I've studied infant GI tracts, including recent observation that we found that babies with colic have a different microbial population compared with babies that don't cry more than 3 hours a day, and elevated calprotectin. So we think this is very important early in life, and I really enjoyed the presentations today that relate to infants. But my real question is for the entire panel, if anybody knows anything about the microbiome of centenarians, people that live to be 100, do they have a different profile? I don't know how many centenarian doctors there are.
I would just comment that what would be interesting, I mean, the problem is that centenarians are old, right? And that's a problem because it's too late, probably. The idea would be to have the centenarians' earlier microbiome to predict.
Thank you.
So I have a question for Scott. So if my memory serves, because I haven't been following the literature as much, but some of the HDAC inhibitors in the past, in the clinical trials, have not done really well. So do you think that butyrate may have, like other actions of butyrate may be much more important in this particular model that you have?
Yeah, I think butyrate likely has a lot of functions, and that's one of the challenges associated with it. And there's even different different people have different opinions on the importance of the mechanism of action. And so I'm always pretty careful to, although I'm more focused on the HDAC inhibition mechanism, I'm the first to admit that it also functions as a ligand for G protein coupled receptors, and various groups have shown that that is an important mechanism, including for cancer. And then a number of other groups have shown that butyrate can actually improve barrier function, and it can also, if given to either naive T cells or even dendritic cells, it can direct them into a Treg cell fate. And so that's also going to be important for cancer and other disease states. Butyrate's obviously been implicated in a lot of things, not just colorectal cancer. And so, those immune functions are going to be really important. And I'm especially curious about the Treg cell function, because on the one hand, that might be tumor suppressive if it is decreasing overall systemic inflammation. But on the other hand, if butyrate is increasing Treg cells in the tumor microenvironment, it could be immune suppressive and give rise to a more aggressive tumor phenotype. And there have also been some reports of butyrate actually having opposite effects, and that could be one way it's doing it. So yeah, that's one of the challenges, I think, that we're still kind of scratching the surface. We know a lot of microbial metabolites, and just focusing on butyrate and a few others that have been studied a lot of them are pretty pleiotropic and act by multiple mechanisms. And so trying to say...
I have a question for you also. Can I ask a question? Yeah. So in your synthetic crypts, do you think that system can be useful to study the function of bacteria that attach to the epithelium?
I think so. They do make mucus also. So an important question in a device like this, an organoid like this, is to have a sufficiently thick mucus layer. And so we do have mucus. We're doing more careful measurements right now to see if the thickness of the mucus really is, because in vivo you're looking at 100, 150 micron thickness of mucus layer. So we're pretty close to that. So I do think it's going to be amenable for looking at microbiota associated with mucus layer, and some that can even translocate inside. It's not the most high throughput device as you might expect. We do have a way of converting this to 2D cultures. You lose the polarized organization, but for some screens it could be useful. And I should point out that people in the audience, the Thad Steppenbeck's group has done some really nice screens with microbial metabolites on organoid technology, and he's made some big contributions on butyrate, and he's identifying other compounds too. So I do like the idea of organoids as a way to screen microbial metabolites for function in a medium throughput manner.
Thank you very much, and let's give a round of applause. Is it okay to get back at 11 o'clock? 11? Yeah, but 11 is okay. Okay, 10:55.
Okay, I'm Dan Xi, a program director and from the NCI. I'm the moderator for this session. It's focused on the microbiome for the treatment. The purpose of this session is to highlight concrete examples of microbiome-based treatment for specific diseases and their outcome, new and novel microbiome-based interventions. We have 3 outstanding scientists here. You will hear their innovative intervention and proactive strategies. So our first speaker is Eric Palmer from the Memorial Sloan Kettering. His topic is microbiota-mediated defense against antibiotic-resistant infections.
Okay, thank you, Dan. And I also want to thank Lita and Ryan for the invitation to speak amongst these really spectacular speakers and presentations. So I'm going to talk about some of the work that has already been partially covered by Rob Jenk on the microbiota changes that occur in patients undergoing bone marrow transplantation. And this was a study that we published quite a few years ago now, a longitudinal analysis of microbiota composition in 5 patients undergoing bone marrow transplantation. And I think what really surprised us at the time we did this study were the dramatic changes that we saw from one sampling to another. And here we were going with weekly sampling in these 5 patients, while some patients, patients C and D, seem to maintain their microbiota quite well. We were able at that time to start to make correlations between changes in the microbiota and clinical outcomes, as well as clinical interventions and their impact on the microbiota. Now, in more recent years, what we have been doing is increasing our collections from these patients, and so we're now quite routinely collecting daily fecal samples from our patients undergoing allogeneic bone marrow transplantation. And the other thing, I just slipped this slide in here to make a point that was something that had bothered us a bit during our previous studies, where we just showed the proportions of bacteria and everything was on a 100% y-axis, to actually quantifying bacterial density in the fecal samples by quantitative 16S PCR, and then overlying on those bars the composition. And I think what this shows is that when patients come in, they have a lot of complexity, but they also have very high bacterial density. And then once we start hitting them with antibiotics, and here's piperacillin tazobactam or Zosyn, which Rob just talked about, has a very dramatic effect, not just on the composition of the microbes, but also about a 6-log decrease in the density of bacteria in the fecal samples that we're collecting. And that really has to be taken into consideration when we think about the changes that are occurring here. Now, I just want to review very, very quickly a study that we published also a few years ago, where we had collected fecal samples from a cohort of 94 patients undergoing transplantation. And we had collected fecal samples on these patients at about the time when their immune system was starting to redevelop. So this was a time of engraftment. And we reasoned that this might be a very important time, given the importance of microbes in the intestine and immune differentiation, that the composition of the microbiota may have a particular impact. And so we ranked patients from top to bottom according to their microbiota diversity. And you can see lots of different colors here. I'll point out the pink, which is Blaudia, which Rob also mentioned just in his talk. And you can see that as you go down this list here, that Blaudia disappears. And by the time you get down to individuals with very low diversity, you're also losing the anaerobic compartment. And at the time we had collected these, we had sequenced them. We had already 3 years of follow-up, clinical follow-up. And so we could ask what impact microbiota diversity at this one single time point had on outcomes. And these are the timelines that are shown here. Open circles indicate that the patient is alive, and the black dots give the date of death as well as the cause of death. And when you plot this just for either graft versus host or infection-related mortality, what we found was that the high diversity individuals had a mortality of around 8%, while individuals who had low diversity had a mortality of 52%, attributable either to GVH or to infection. And that this difference in mortality actually accrued over the course of 2 years following this single time point measurement here. And that suggested that the composition of the microbiota may be having a long-term impact, the composition at the time of engraftment. And so that led us a couple of years ago to propose to perform a randomized trial of what we call autologous fecal transplantation. And what we do is we collect fecal samples from patients, we characterize them, we freeze them, and then we wait for the patient to go through bone marrow transplantation. We wait for them to engraft, and then about a month later, we thaw the sample and we randomize the patients either to get their own microbiota back or to undergo routine management and not get their microbiota back. And at this point, we've screened a lot of patients. We've randomized 51, 26 have gone to the treatment arm and 25 to the control arm. And we've been able on 25 patients now to look at the effectiveness at a microbiological level of FMT. We've collected fecal samples before, after FMT. And because, as Rob pointed out, we've sequenced now lots of different microbiotas from our bone marrow transplant patients. We can plot them on these t-SNE plots and put any particular sample within a region of either diversity, Streptococcus domination, Enterococcus domination, or lacto or staph domination. And so this allows us now to look at each of the patients either in the control arm or in the treatment arm to see where they started and where they ended up following randomization. And what we find is that the treated patients, by and large, end up back within the diversity region, and they also have very high percentages of reacquisition of their starting, of the microbiota that they came into the hospital with. This is perhaps a bit more simpler way of looking at things. This is looking at representation of all these different bacterial taxa. Each horizontal line is a patient. And we can see that pre-transplant, they have representation of lots. Post-transplant, they've lost many of these species. And following FMT, many of these are regained. In our control patients, we see that many of the losses that have occurred persist following transplantation. Now, we're at the midpoint of this trial, and we do have follow-up now on many of these patients. This is our most recent data, where we're showing the 26 patients who've undergone FMT, and these are our control patients. Here we're documenting bloodstream infections, which all of these bloodstream infections here occurred in one patient who ultimately ended up dying of graft-versus-host disease. We have one death in the treatment group due to disease progression, while we have 3 deaths that have occurred due to infection at relatively early time points in our control population. So this trial is continuing to accrue. We've also looked at things like viral reactivation, and we see actually in both groups quite a bit of viral reactivation, although the severity, at least at this point in the trial, seems to be somewhat greater in the control population. Okay, I'm going to jump around a little bit now because I want to cover several areas where we have tried to identify bacterial species that confer resistance against some of the major pathogens that we encounter in our hospital. So this is a paper that was published a couple of years ago now, where the strategy that we used was to treat mice with different antibiotics and then to see how susceptible they were and how long were they susceptible to C. diff infection. And what this strategy allowed us to do was to sequence the microbiota of mice, in this case, that were either susceptible or resistant to infection, and to begin to correlate the presence or absence of specific bacterial species, commensal species, with susceptibility to infection. And we ended up with, using the Spearman rank correlation, a number of different species that were highly correlated with resistance to C. diff infection, particularly in mice, and that's shown in red here, where the diversity was very low, but the resistance was very high. And so that pointed out an organism in particular that jumped out, Clostridium scindens, was highly associated with resistance. What Charlie Buffy, an MD PhD student in the lab, was able to do was to administer Clostridium scindens to mice, and it reduced susceptibility, increased survival. But in particular, when we paired Clostridium scindens with these other organisms, Blaudia, Pseudoflavanofractor, and Barnesiella and Testa hominis, we could get 100% survival, marked reduction in C. diff growth, as well as toxin production, and suggesting at least that some of these other organisms here were facilitating the ability of Clostridium scindens, which we know modifies bile acids into a form that makes them more inhibitory to C. difficile growth. Now, we use a similar strategy again to try to look for bacteria that specifically confer resistance to vancomycin-resistant Enterococcus, which you learned about yesterday. Here, we know that if we treat mice with ampicillin, that we can make them very susceptible to VRE, intestinal domination. If we dilute normal microbiota, we thought that we might reach a dilution where some mice would be protected and some would be resistant. And in fact, when we diluted to 10 to the minus 6 and administered it to mice, we saw that some were fully resistant, some were fully susceptible, and some were in the middle. Sequencing now the composition here, we again came up with a list of organisms that Sylvia Caballero worked with. She then spent a couple of years trying to grow some of these bugs, and finally ended up getting a consortium of 7 bacteria that we could administer to mice and demonstrate high-level resistance to VRE colonization. So that was exciting. Ultimately, she was able to show that Blaudia productive Clostridium boltii, Parabacteroides dissonis, and Sartori were the 4 bugs that, when administered together, dealt with this high-level resistance. Now, we wanted to get at a mechanism by which these organisms are conferring resistance, and what we found was that co-culturing Blaudia productive, but not the other 3 organisms, strongly inhibited VRE growth in co-culture. And in fact, this inhibition was mediated by the culture medium that Blaudia had grown in. So we knew it was something that Blaudia was releasing. Now, and it turned out that it didn't just inhibit VRE, Ephesium, it also inhibits Faecalis, a bit Staph aureus, and also Listeria monocytogenes most effectively. So then, we wondered, is this something that's true of all Blaudia species? And we were lucky to have from Kenya Honda's collection another Blaudia producta that we tested, and surprisingly, it didn't inhibit VRE growth at all. And that was a bit of a mystery, and so we went to our sequencing facility and we sequenced both strains and then did a genomic comparison. And what we found using these software platforms to look for bacteriocins, that our strain, the Sloan Kettering Blaudia producta strain, actually had 4 loci that encoded for bacteriocins. And there was one in particular that was a complete operon for something referred to as a lantibiotic. And these lantibiotics are fascinating molecules that are produced by a number of gram-positive organisms. The best known one is called nisin, and what you can see are all these strange lanthionine bonds that are formed actually by the bacterium. The Blaudia producta sequence is different, but very similar in structure to the nisin molecule, and is what we suspect is mediating the inhibition of VRE. So along very similar lines, we've now also recently started to look at resistance to an enterically acquired pathogen, Listeria monocytogenes. And we were very surprised. People say that mice are intrinsically resistant to enteric infection with Listeria. But it turns out if you treat them with streptomycin, you can actually get mice highly infected with disseminated Listeria infection. And again, it correlates with a loss of a variety of anaerobic organisms. And Simona Beccattini recently was able to assemble 4 strains that were most inhibitory and that provided very high-level resistance in vivo against Listeria infection. And so these are now 3 circumstances where assembly of obligate anaerobic organisms can provide this high level of resistance. So I wanted to just point out my critical gap. I think that key gap is incomplete representation, characterization, and open and unfettered availability of human microbiota-derived commensal bacterial species. And I think that this could be a tremendous resource, a biobank or a biorepository that we could go to so that Peter Turnbaugh doesn't have to worry about growing 20 or 30 species of Blaudia to take the next step, but just to be able to order them. I think they should be genome sequenced. We should know their metabolic dependencies, their metabolic products, short-chain fatty acids, bile salts, vitamin synthesis, and potentially be tested in germ-free mouse models, in my case, to determine colonization resistance. And finally, I just need to thank, these are I consider them kind of the founding fathers of our microbiota project. You see Rob Jenk is still on here. I can't get myself to take him off, even though he's escaped to Houston. And these are the fellows who have done much of the work that I presented. Thank you.
Thank you very much. So our second speaker is Dr. Timothy Lu from Massachusetts Institute of Technology. His topic is engineering biological computers for human health application.
Great. Well, thank you very much for the invitation to be here. It's an honor to tell you about some of the work we've been doing to try to use the tools of synthetic biology to engineer the microbiome. So our lab is interested in trying to develop tools that allow us to perhaps more precisely sense and respond and manipulate what's going on in the microbiome. I think one of the driving reasons for this is we sort of realize that in order to be able to do precision changes to the microbiome, we need new techniques to be able to either delete specific members of the complex microbial community in a targeted way rather than sort of using broad spectrum antibiotics, which we've heard have many complicating factors over the course of this workshop, as well as can we actually introduce specific microbes into the gut that actually allow us to detect, perhaps in real time, what is going on inside of a complex microbial community like in the gut, and then be able to respond by trying to treat disease. So that's what I'm going to tell you about today, several technologies that we've been working on in the lab to try to enable such research going forward. So I want to start off first on the subtractive side. How do we build perhaps more narrow spectrum antimicrobial strategies that allow us to perturb members of a community? So we've been for a while quite interested in this problem from the perspective of trying to treat infections, which is that can we move away from the use of very broad spectrum antimicrobials and try to get to the point where we can develop very targeted antimicrobials that you can readily reprogram to go after whatever organism you want. One of the associated challenges, which I won't talk in much detail about, is if you have narrow spectrum antimicrobials, for them to actually be practically usable in the clinic, we also need rapid diagnostics that can actually allow us to know which antimicrobials to be used in the clinic. Fortunately, there are a lot of folks trying to develop more rapid diagnostic tools these days for microbial infections. So I'll focus more on the latter. How do we actually think about designing targeted antimicrobial agents? So we've been exploring a couple different ways to do this. One, using engineered antimicrobial peptides. Secondly, using engineered bacteriophages. But today I want to tell you briefly about some of the work we've done to try to incorporate the CRISPR nuclease technology as a way of editing bacterial communities. And so the idea is, can we actually develop antimicrobials that don't target bacteria based on conserved mechanisms, but rather allow us to build antimicrobial agents that target bacteria at the level of DNA, which using the CRISPR system, we might be able to very readily reprogram against. And so I think everyone in the audience knows about this system, but just briefly mention, at a rough basis is basically trying to turn the Cas9 nuclease or any other programmable nuclease into a sequence-specific cleavage system where you can then redirect the activity of that nuclease against a target in the genome. And the idea in bacteria is that if you could sort of repeatedly cause cleavage at a specific target site, like a virulence gene or an antibiotic resistance gene, could you basically force that bacteria to die or mutate that sequence? So one of the strategies we have to develop is how do you actually get this thing delivered? One strategy we developed was to basically encode the Cas9 system onto a phage-mid, which we could then package, sort of produce in the lab, and then throw onto our bacteria of interest. So the idea here was that the bacteriophage would then deliver the Cas9, which we can then reprogram to go after a specific target. So for example, here we designed the Cas9 system to go after a genomic target. In this particular case, it was a point mutation in the gyrase A gene that confers quinolone resistance. We have two engineered phage-mids here, one that targets the NDM1 gene, the other one that targets the specific gyrase A mutation. And so when we put these two phage-mids against the wild-type population, which are actually both susceptible to phage infection, but you actually don't get any toxicity against these, and it's because they don't actually contain the target for the Cas9. But if you actually put this onto the antibiotic-resistant version of the E. coli, you can get about four-order magnitude drop in the viability of those cells with the specific Cas9 system, indicating that potentially now we can use this method to use sequence-specific nucleases to try to destroy particular bacteria while sparing the rest. In this particular paper, we actually showed ultimately that depending on how you configure the Cas9 antimicrobial, you actually generate different effects. So you can target genomic locations, as I mentioned. If you could target things on plasmids, which many of these antibiotic resistance genes end up sitting on, if those plasmids have addiction systems or toxin antitoxin systems, you can cause those cells to die. In other cases, if in the absence of those, you can actually cure the cells of those particular plasmids without actually affecting cell viability. Ultimately, where we're going with this is to try to demonstrate that we can actually use this to edit more complex communities. So here's an example where we made a synthetic three-member community composed of the blue, purple, and yellow bacteria and engineered a CRISPR system that only targeted, for example, the yellow bacteria in this case, or another system that only targeted the blue bacteria. And the idea is, even though all three of these bacteria can be actually productively infected by the phage-mid, only one of the three actually gets killed based on a sequence-specific nuclease. Now, as you imagine, one of the key challenges here is really trying to deal with the delivery issue. How do you get these systems delivered efficiently? And so in the lab, I won't, just because of time, I won't talk about this in a lot of detail, but we've been developing libraries of phages now that can mediate more efficient delivery into a wider spectrum of bacteria. Alternatively, we've been exploring conjugated plasmid strategies. Normally these bacteria do horizontal gene transfers, as we've heard over the course of the few days. If you could piggyback upon that mechanism and sort of have a gene drive for bacterial systems, this could be another way for us to think about editing bacterial communities. All right, so I talked about the subtractive techniques. I want to spend the rest of the time telling you about some of the work we've been doing on engineering bacteria that could be delivered into the gut for a variety of different applications. So the ultimate idea here is very simple. Can we genetically modify bacteria and deliver these into the gut through an oral fashion? And we've been exploring two different applications here. The first, can we actually engineer sensors or diagnostic bacteria that actually detect biomarkers of disease and then report that out to the real world? And then secondly, can we take those bacteria and then engineer them to actually provide treatment locally in the gut? So can we do sense and respond type behaviors? To do this, we actually spent a lot of time doing the genetic engineering of these strains. So we spent a lot of time sort of as engineers getting really efficient at modifying these bacteria. So I'll show you some of the projects that are ongoing in the lab. One project that we've been doing together with Anantha Chandrakesan and Phil Ndu in electrical engineering at MIT is to actually co-house engineered bacteria that actually detect biomarkers of disease onto a little pill that then transmits that information wirelessly from the inside of the body to the outside. So the idea here is, could you swallow a pill, but instead of this being like a pill camera, what if you had some sort of front-end sensor, in this case a bacteria, but in other cases you could think about sort of cell-free systems that allows you to sense in near real time what's going on inside of the gut, transmit that to the outside world. So in this particular case, we spent a lot of time sort of optimizing the microelectronics so that we can package everything to a small battery form factor, but also engineering the bacteria to sense the specific biomarkers of interest and produce a detectable signal. In this particular case, we're actually producing bacterial luciferase. So here's just a picture of the little pill. It's about 3 or 4 centimeters in size. You can swallow it. And basically, the first instantiation of this was to actually show that we could detect something like bleeding inside of the gut. So we engineered a probiotic bacteria that actually sense heme and then produces luciferase as a response. And so here you can see in the presence of blood, as well as the sensor, in about 15 or 30 minutes in this in vitro case, the circuit turns on. If we make a deletion of one of the components in the cells, basically the system stays off. Now, this is done in vitro in a test tube. We've also been working now with Giovanni Traverso in the lab to try to test this now in a pig model of bleeding, where we take anesthetized pigs and then basically give them blood or not. And then we test whether the chip is actually able to then detect bleeding in the gut. And here's just some of the preliminary data we've generated. This is off the press yesterday, where we've just generated some of these pills, put them into the pigs, and shown that indeed with the sensors we can detect in approximately 45 minutes the presence of bleeding in the gastric system versus the negative control. So, we're continuing to optimize this, but I think there's promise here, and once we have the system up and running, it should be relatively straightforward for us to then adapt the front-end sensor component to detect other metabolites of interest, and then have this as a real-time reporter where, this pill could be, talking to your Bluetooth connection on your phone. All right, so a lot of the work that we've done so far has been in E. coli. One of the things we'd want to do now moving forward as part of this field is to really try to genetically domesticate a lot of the really cool organisms that we've heard about over the last few days. The challenge when doing that, first, you got to be able to grow the organism, but I think a lot of folks are trying to do that. But secondly, making those organisms genetically tractable to the point where we can actually do genetics in them to study their function is a challenge. So a few years ago, together with Chris Voigt, we decided to see whether we could actually port a lot of the tools that we're used to using in E. coli genetics into an organism like Bacteroides. So we've now imported a lot of these tools into a variety of strains of Bacteroides, as I'll show you, and now have the ability to introduce genetic circuits and actually build sensors and even therapeutics in this context. Basically, what we spent a lot of time, this is a little bit boring from a genetic engineer's perspective, but actually really important, which is to build really large libraries of gene regulatory parts that we sort of take for granted when we're working with other organisms. So we spent a fair amount of time building promoter libraries and ribosome binding sites that in combination can allow us to regulate gene expression over about 5 orders of magnitude. We built 4 different inducible systems that can be turned on by different metabolites, as well as importing the CRISPR-I system into this organism so that you can do functional genomics, genetic studies on what genes lead to what function. So here's just some examples. We basically have a rhamnose inducible system, a conjoined sulfate inducible system, and a rabinogalactan inducible system, as well as the IPTG system ported over from E. coli, allowing us to control gene expression in orthogonal ways. And so, for example, if you cross all 4 of the inducers here with all 4 of the promoters, you can see we get pretty orthogonal induction. So this gives you sort of 4 little knobs by which you can tune the expression of genes in Bacteroides now. We've also incorporated what we call memory circuits into these cells. So imagine eating a Bacteroides, having it sense what's going on in your gut, and then when it comes out the other end, if you could collect those Bacteroides and sequence them or do some sort of simple nucleic acid test to see what actually happened to the Bacteroides while it was going through your body. So one of the ways to do that is to use a class of proteins known as integrases or recombinases. what those proteins do is they basically flip a piece of DNA upside down. So, for example, if we add rhamnose to these cells, it induces the expression of the integrase. It recognizes the ATDB and P sites that correspond to that recombinase and flip it upside down. Now, simply now by sequencing and looking at the orientation of that DNA, you have a very simple 0 or 1 bit, just like in our computer. And so by adding more rhamnose, for example, we can switch on the memory circuit and get near 100% writing of that particular memory unit. And the circuit basically turns on within about 2 hours or so. We've now moved on to show that these circuits can actually work when you colonize mice with these strains and then feed them different inducers. So, for example, we've taken mice here and colonized them with beat theta and then collected their stool and basically looked at whether their stool is glowing or not. So here in this particular example, we fed the mice arabinogalactan from day 2 and day 4. And in this simple system, you're just turning on luciferase expression. And we show that you can indeed switch on about 2 orders of magnitude of luciferase. And then after you take away arabinogalactan, it goes away. And we've also done the opposite. So using CRISPR-I, we can use IPTG, for example, to regulate the CRISPR-I system and shut off a reporter. And again, from day 2, day 4, when we feed the mice IPTG, they indeed drop the expression of luciferase, and it recovers once you remove that from their food source. So essentially what we've been doing is building up these sort of tools in the lab, and we're continuing to push forward to try to provide these as a resource to the community so that you can introduce sensors into a microbial space. You can genetically modify more and more organisms to do functional genetics on them. But ultimately, our interest is trying to push these technologies into the clinic. So I'll tell you a bit about some of the work we've been doing on that end. So about a few years ago, we had spun some of the technologies out into a company called Synlogic that was now trying to use these circuits as a way of treating disease. The idea here is to take some of the genetic circuits we've devised and introduce them into, for example, a probiotic strain that can be consumed orally to try to treat metabolic diseases. I'll show you. So the approach that we've taken with this is actually to try to develop these engineered probiotics to try to treat rare disorders, including inborn errors of metabolism. 2 of the ones that we're focused on, urea cycle disorder, where the problem is basically you get toxic buildup of ammonia in the bloodstream, as well as another disease, phenylketonuria, where patients don't have the ability to metabolize phenylalanine. And so we're in the phase 1 trial right now with the UCD program, and PKU should be coming hopefully at the beginning of next year. And we're also developing preclinical candidates to go after some other disease types. So here's just an overview of why we think this is an interesting approach. You know, what we want to be able to do is give patients an oral dose of an engineered probiotic and have that be able to lower systemic ammonia levels. So instead of having to inject yourself with an enzyme replacement therapy or do gene therapy, what if you could actually just do this through a non-invasive oral route? If this worked, this could also potentially have benefits in populations where patients have acquired liver disease and can't process ammonia properly. So the basic approach is, as I mentioned, very straightforward. We take an engineered probiotic like E. coli Nissl, which has been used in the clinic as well as sold over the counter for quite a while, and we genetically modify that organism. Here, in particular, we sort of amp up this pathway for consuming ammonia and making arginine. It's about a 10-member pathway that you have to genetically modify to really get very efficient conversion. And the idea is, if you feed this orally, can you basically convert the ammonia load in the gut to arginine and have that sort of flushed out as over time. We've also put this circuit under an anaerobically inducible switch. And this was done for 2 reasons. One is to make sure that this thing is only really on at very high levels inside of the gut. And number 2 is also for manufacturing reasons. These bacteria are genetically modified to be really, really efficient at doing something that doesn't really help them at all in terms of surviving. And so we actually need a switch to keep them off during the manufacturing phase so that we can make a lot of it and then switch it on before we give it to the patient. Here's just some examples of the particular strain. So if you look at E. coli Nissl in vitro and its ability to produce arginine is quite low, but the 2 genetically engineered constructs here are very, very efficient now at being able to convert ammonia into arginine. And then in a SPF-ASH model of hyperammonemia, where you give mice high protein diet and look at their systemic ammonia levels, you can see normally if you're on water, a normal chow, you have a relatively normal blood ammonia level. On high protein chow, it elevates. But when we treat these mice and just basically feed them the engineered probiotic, we can drop that back down to the systemic levels. So as I mentioned, this is now in the phase 1 study. We're very hopeful to get readouts of this and see how this particular strain performs. We also have other work going on in several other areas, including PKU. And I think this is an exciting foray of some genetically engineered probiotics in the clinic. So with that, I just want to finally just address some of the critical knowledge gaps that we're trying to address with some of our technologies. I think we all want to move forward and try to understand the functional interactions between microbes as well as microbes and the host. And I think in order to be able to do that efficiently, we actually need better sensors and better tools to be able to actually see what's going on in more real time in specific spatial locations and in different bacteria. And so we're trying to develop key resources and tools for that. I'd also like to echo the previous speaker's talk around, we need better collections of these organisms that allow sort of the whole community to be working on the same set of organisms, preferably really well-characterized ones that we can, build up good libraries, characterize them so that they're also available for the community to be able to use. And so with that, I'd like to just summarize with sort of this slide, in that we're trying to develop these sort of tools to solve this particular set of problems. And finally, thank the people who did the work. So Rob Siderick and Mark Mimi were the guys who developed the targeted antimicrobial approach. And Mark, as well as Chris and Alex, were the folks who did the work on the engineered Bacteroides, as well as Mark is working with Anantha on the engineered microbial sensors. Thank you very much.
Okay, thank you very much, Dr. Lu. Yes, it is very interesting. So we have the last speaker of this session, Dr. Eugene Chang from University of Chicago. His topic is connecting the gut microbiome and the cancer through circadian rhythm.
Thank you, Dr. Shi. So I want to thank Lita and the organizing committee for including me in this stellar cast of speakers over the past few days. But I also want to thank the audience for sticking it out to the end. And, one of the things about being very last on the last day of a program is that it's sort of an undesirable position to be in, but I'm an optimist. And, when they actually talk to audiences after meetings, they ask, well, what do you remember? And they remember what was said in the beginning and what was said at the end. So I'm hoping that, you guys are like the Trump core supporters, no matter what I say, you'll remember what I say. So I'm going to talk about a topic that is a little bit offbeat for what I do, but it does connect a few things, and it is germane to the theme of this session. And that is connecting the gut microbiome and cancer through circadian rhythms. And there is some logic to this, and I'm just going to go through this first introductory slide, which is that, there are three parts to this story: cancer, metabolism, and the intestinal microbiome. And I think there's a large amount of literature that connects cancer to metabolism and microbiome to metabolism and intestinal microbes to cancer. And often these are bidirectional pathways. But what I'm going to do today is provide an example of a pathway that starts with the intestinal microbiome, and through this diurnal variation that we and other groups have observed, altering host metabolism by affecting circadian rhythms, which do have some relevance to cancer biology. So how do we begin in this story? Well, we began with a very simple observation that many people have made, we had noticed that germ-free mice are very resistant to Western type of high-fat diet. And that's just shown here on the left. If you put SPF mice on a high-fat diet, they gain weight in comparison to their counterparts on low-fat diet, which is shown in the blue. But if you gave the exact same diet to these mice that are germ-free, they actually don't gain any weight. They're resistant to these high-fat diets. And this would suggest that somehow gut microbes are very important in in the metabolism and what we do with the calories that we eat. Now, another interesting observation is shown in the left panel, and that is if you look at the germ-free mouse on a regular chow, this mouse has to actually eat more per day in order to maintain the same amount of weight. And you can see that it's much more than the other three groups. And so what this suggests is that somehow, without having gut microbes, the metabolism of the host has been altered. So we wanted to drill down on this a little bit better. And we did a very simple study, and this was to look at the liver transcriptome and compare it between the germ-free mouse and the conventionalized mouse. And why the liver? Well, the liver is your major metabolic organ. So what we did was we did a transcriptomic analysis, and it turns out that there are thousands of genes that are differentially expressed between these two groups. And if you do the pathway analysis, what was really eye-opening, at least for me, was that 2/3 of these genes are related to metabolism. Only 1/3 was related to immune mechanisms. And for me, it really began to underscore the potential relationship between gut microbes and our metabolism. Now, if you drill down on that data and you look at the pathway analysis, what comes at the very top, where many of the members are included in these networks, are circadian clock genes that are involved in regulating circadian rhythm. Now, when we saw this at first, I didn't know anything about circadian rhythm, and I wasn't sure what this meant. But after reading about it, it actually made a lot of sense. So circadian rhythms are basically controlled by a series of so-called clock genes. These genes, for example, include inducers like clock and BMAL, which activate through transcription a number of gene pathways that ultimately are shut off by repressors like CRY and PER, which do it through a translational regulation. So the bottom line is that you have this oscillatory behavior through the course of the day, which is basically like an on and off switch. And what it does is this kind of core loop regulates a series of genetic pathways that ultimately will control peripheral circadian clocks in a way that basically tells our body when to burn energy and when to store it. And most of this is driven by light and dark signals. That is, when you wake up in the morning, your circadian switch goes on and says, okay, you got to burn energy because you're going to use it. And when you go to bed at night, you actually start to store it. So it's very important that you have this kind of regulatory system, and it's very inherent to almost all life forms. Now, it turns out that circadian rhythms are more than just regulating metabolism. And they regulate, for example, behavior, waking and sleeping. But relevant to the discussion today is that it turns out that circadian clocks are very important for cancer biology. And this is shown here. This is an excellent review by the Sansoni-Corsi group. And I seem to have lost my pointer. Oh, no, here it is. This is just a very broad generalization of how circadian clocks are relevant to cancer. It's known, for example, that epigenetic regulation is very important for circadian regulation of transcriptional events. And this occurs through acetylation and methylation of histones, DNA methylation, microRNA, and also recruitment of other transcription factors. But another point that circadian rhythms are important is that some of these circadian clock genes, for example, PER and CRY here, interact with a number of key proteins that are involved in cell cycle regulation. And other targets include things like tumor suppressor genes and other regulators of cell cycle. So not surprisingly, if you disrupt circadian function, as shown here, and this can occur through many different ways. For example, it could be through sleep disorders, shift workers, who, by the way, epidemiologically have been found to be at increased risk for cancers, to experimental systems where you can pinpoint specific disruptions of the circadian clock. And there you can actually see that you will affect both tumor genesis as well as progression of tumors. So there is a link between circadian rhythms and cancers, and it occurs at multiple levels. Now, how is this all relevant to the gut microbiome? Okay, so I'm going to go back to this germ-free model. And in this instance, what we looked at is, okay, we knew that there was some kind of metabolic abnormality of these mice. Actually, maybe a metabolic advantage since they can eat all they want and they never get fat. But on the other hand, what we did was we asked a very simple question. Well, what happens to their circadian rhythm under both high-fat and low-fat feeding conditions? And we looked in the brain. So the master regulator circadian rhythm is in a place called the suprachiasmatic nucleus, which is located in the medial basal hypothalamus. And then we looked in the periphery, and we looked at the liver specifically, which is your major metabolic organ. And what you can see is that high-fat diets do alter circadian behavior, in this case shown for this one inducer, BMAL1. And shown in the blue is the comparison of what this rhythmicity is for BMAL in a low-fat fed animal. And so this change is actually not surprising. It's been reported by many groups. In the liver, the changes were not as dramatic between high-fat and low-fat diet, suggesting that this regulation does have differential effects depending on where the circadian clock genes are located. But the most striking thing, I think, is shown on the right panels. And this is data from the germ-free mouse. And what you can see here is it doesn't matter whether it's high-fat or low-fat diet, something has happened to the amplitude as well as the rhythmicity of, for example, this BMAL1. So this is seen in other circadian clock genes as well. And I think that you can see that this effect, it occurs both in the central as well as in the peripheral regions. Now, this was really profound, at least to me, because it was always thought that light and dark stimuli were the major drivers of circadian rhythm. But what this study showed was in absence of a gut microbiota, those light and dark signals are not getting through. Okay, these mice were under the same light and dark conditions as their SPF counterparts. So this really underscored the importance of gut microbes in regulating our circadian rhythms. It wasn't just necessary, but it was absolutely essential for normal circadian function. So what happens on the microbe side? You know, circadian biology is all about time. So what happens to the microbiome over the course of 24 hours? And this is shown here in a PCOA plot. So on a low-fat, fairly diverse diet, you can see that there are changes throughout the day. So Zeitgeber 0, Zeitgeber time 0 is actually when the lights are turned on, and Zeitgeber 12 is when the lights are turned off. And you can see that there is a fluctuation of these populations over the course of the day. That behavior is lost when these animals are placed on high-fat diet. Not only are the oscillations lost, but there is a shift in the membership of these two populations. So what's the consequence of this? Well, the consequence might be that some of these metabolites that microbes make, particularly the ones that were oscillating through the course of the day, which tend to be mostly fermentative organisms, might have some effects on our own biology. So just to ask the question, well, what's happening to some of these metabolites? So I showed two of them here. One is butyrate, the other is hydrogen sulfide. So you can see that butyrate has nice oscillations over the course of 24 hours that's seen with a low-fat diet. But on a high-fat diet, which is a milk-fat type of diet, you see that the levels and the oscillations of butyrate are fairly attenuated. Now, if you look at hydrogen sulfide, you see the opposite. So on a low-fat diet, the oscillations are modest, but they seem to be accentuated in a high-fat diet, and the amplitude seems to be higher. So what is the impact of these metabolites? Well, again, we're looking at acetate, butyrate, and hydrogen sulfide with the surrogate sodium hydrogen sulfide. And on the left side of the panels, we're looking at PER2, and the right side, we're looking at BMAL1. And if you look at the middle line here, you can see that butyrate given to these hepanoids, these are hepatic cells derived from stem cells that we derive, and they grow as organoids. And when you expose them to butyrate, you can see that there's a very profound shift in their phase. There's a phasic shift, which is seen with both PER2 and BMAL1. It's very specific because you don't see it with PER2, with acetate, or with hydrogen sulfide. And one of our collaborators, Jen Evans at the Medical College of Wisconsin, also looked at the effects of butyrate on brain slices from the PER2 luciferase transgenic mouse. And again, what she saw was that when she adds the butyrate here, there's a almost 180-degree phase shift, which you can see quantitated here. So this is very profound in terms of chronobiology. To summarize the findings up to this point is that on a low-fat diet, what we see is a nice oscillation of microbial populations and metabolites that provide important cues for normal circadian function, and that leads to states of leanness. In high-fat diets, in contrast, these microbial oscillations are lost, leading to aberrant microbial signals and affecting central as well as peripheral circadian function to lead to states of obesity. In the germ-free mouse, we lose this transducer, and in essence, there are no microbial signals, and that causes disruption, but in this case, to a default pathway of leanness. So I end just with one slide, which suggests that maybe we can leverage this knowledge to a therapeutic benefit. So this is a study where we did administration of butyrate by an intraperitoneal approach in mice that were in a high-fat diet. And what you can see is that we have circadian disruption with this high-fat diet, but when we give butyrate at a particular time of the day, we can correct it, and that's associated with an improvement in the accumulation of fat and gonadal fat pads. So I'm going to just end with what I think are the current gaps and challenges and also opportunities. So the gaps are inadequate understanding of mechanisms connecting the microbiome, metabolism, and cancer. The challenge is developing better tools for identifying microbial and metabolic oscillators. And then I also think that another gap is that humans are very difficult to study, particularly with circadian biology. And this is I think Gary Wu brought this up yesterday. This is really an obstacle when you're trying to do time sequence collections on a high resolution scale. How do you do that? The opportunities are developing microbiome-based predictive markers and interventions for the prevention and treatment of cancer through this kind of circadian link, and then going beyond description in human studies so that we can develop effective and sustainable therapeutics. And I just want to end by acknowledging all these people. It's a team and involves multiple institutions from many disciplines. But I particularly wanted to point out Vanessa Leone, who's really been the heavy lifter in these studies. Thank you very much.
Thank you very much, Dr. Chen. It is very provocative. So now we open for the question. And please identify yourself.
I have a couple of questions. One for Eric. So this is very exciting, the Blautia story. The question is, have you done experiments to beyond the in vitro? The question is, do the in vivo, if this is a major factor for mediation of VRE resistance, the peptide, or you don't know yet?
We suspect it is. I mean, we know that the two Blautia strains, one is highly effective at clearance, the other one, which lacks the antibiotic, is ineffective. But as you well know, one of the big challenges right now is to perform genetics or genetic manipulations of these strains, and so we're working very hard to knock out the locus in the Sloan Kettering Blautia, and we're starting to have some success with that, and I think that'll answer that important question.
The second question is for Dr. Liu. So the engineering of these strains is beautiful. The sensors are potentially. The only concern I have with this, with that is fine. The concern I have is with the known fact that there is colonization resistance, not just against pathogens, but also against most of the commensal bacteria. We see that, we suffer that in doing experiments in mice and also in humans. So this will rely, so a major potential problem here, you might engineer a beautiful sensor, but then do they provide colonization, stable colonization for years or months? So how you will address that? Because this is potentially is a big problem.
Can you hear me? So yeah, that's a great point. I think right now at least the stuff that we're pushing in the clinic are actually strains that are purposely modified so they don't colonize for FDA or safety reasons. And so these would be things that you would have to take on a routine basis, at least with the current envisioned. I think we're very excited about the potential for moving into commensal organisms that potentially can persist for a much longer period of time, such as the ones that people are investigating in a lot of these studies. I feel pretty confident we're going to have the tools to genetically modify them. I think it's more of a regulatory question of what's the path of being able to use those organisms in people if they're genetically modified.
Yeah, my question's for Eugene. I guess we'll spread them around. I love the circadian part, and of course fits nicely with drug metabolism and circadian patterns as well. Have you tried looking at seasonal effects or things like that? Or maybe in humans, it'd be interesting to look at weekends versus weekdays to see what kinds of other patterns might appear in people's microbial fluctuations.
So we haven't done those studies, Mike, but they have been done experimentally. People have done things like, free running, which is keeping, for example, these mice in conditions of total darkness or total light, or they'll do time shift type of experiments. And I think that they underscore the importance of, normal circadian rhythm and how disruptions of it can be very deleterious to things like cancer growth and development, as well as to metabolism.
So Rob Britton, Baylor College of Medicine. This question's for Dr. Liu. So when we do probiotics and engineered probiotics in mice, I would argue that we kind of eliminate some of the biogeography because we slam them with such a high dose of bacteria and essentially the same dose we give to humans. And of course, there's such a huge area difference. So do you have any sense of what kind of dose you're going to need to give these to people to be effective and make sure you actually encounter the metabolites?
Yeah, I think that's partly what the dose finding is, we need to do in the current trial. I think, and it is in part basically the more potent we can make these particular strains, why we spend a lot of time metabolic engineering them so that we sort of reduce the requirement for numbers. We are at a little bit of inherent disadvantage in that, the E. coli Nissl that we're using doesn't colonize at very high levels, so it doesn't actually stay around at very high levels, and that sort of poses a challenge. I think, the doses that we've put into non-human primates have been up to like 10/11th per dose, and we've been able to look at biomarker studies and see an effect in that system, but we're going to have to see if that replicates in people.
Dr. Shang, Udi Langen, FDA. Did you get a chance to have a glimpse of the link between the immune system
and the gut microbiota driven by this circadian rhythm? The link between the immune system and the gut microbiota driven by the circadian rhythm.
So actually, that's a great question. So work that Laura Hooper has done, and also Vanessa Leone, many systems, innate immunity in particular, is very circadian. It's under strong circadian regulation. And both Laura and Vanessa have data that suggests that that is also important in maintaining this diurnal variation in the microbiome, so that you know, when things begin to bloom, you need to sort of bring them back down. And so there's this sort of seesaw regulation where the bloom of certain organisms actually induce innate immunity, and that then brings the population down. So there is a coordination. I do believe that immunity is important in this kind of cycling and coordination between host and microbe.
Nancy Sorden, medical subject headings at the National Library of Medicine. This has been a great conference, and I find it been very supporting of the community in the challenge question and the gaps questions. We have been putting in microbiome organisms into the medical vocabulary recently, and I would like to encourage people to, if you have any concepts that you think are important to be searchable in the literature, to please let us know that if you go to the National Library site, you'll be able to get to the medical subject heading site, and there's a link where you can make requests for concepts to be searchable. And also, I just, the way that this has been such a supportive community, I just wanted to say that, you know, we are a community of scientists. We're going through life together, and we're supporting each other, and we hold each other very dearly, and we also must hold the rest of humankind dearly. And so I just wanted to applaud Dr. Owen White Wednesday, for announcing the removal of the Confederate statues. You know, these are statues of heroes, of people who believe that it's okay to abuse others and to take away the rights of others. And so I think we need to support when people stand up to say what's not right, and just like we support each other in our careers and in our lives.
Okay, great. I think we are at the end of these sessions. Yeah, let's give a big applause for all the speakers and all the people still here, and Dr. Yelita. Now we go to lunch and the post session.
Welcome. We're wrapping up this workshop with an experiment, which is to, we're all scientists here, to get agencies together. So let me tell you who these people are. All of us served on the Microbiome Interagency Working Group, which is the federal committee under the OSTP's National Microbiome Initiative, okay? And there are 16 agencies, but I asked those representatives from those agencies that have an interest in the human microbiome area of the microbiome field to come and join this joint agency panel. In addition, I asked some particular offices at NIH to join this panel as well, because they don't necessarily always get a voice in these discussions. One office is the Office of AIDS Research. You can go ahead and raise your hand. Or you go in order, but just so you know who the NIH people are. Oh, am I not talking loud enough? I always think I'm really loud. The Office of Research on Women's Health, and the National Institute for Minority Health Disparities. Okay? So those are the 3 NIH offices, but the rest are other federal agencies. And remember, the theme of this workshop is not only to assess where we stand in the human microbiome research field, but to also really, more importantly, identify obstacles, critical gaps, needs, and challenges that we need to address in order to advance this field over the next 10 years. You've heard over 3 days, lots and lots of ideas around what are gaps, needs, and challenges. So I thought it would be valuable to also hear from the agencies that are either currently mounting human microbiome research activities, are going to be funding human microbiome research activities, or undertaking internal studies on the human microbiome. They have a series of slides. They're going to be very quick slides, so you're not going to hear a big, long discussion about the agency. And Cliff is smiling because I was really hard-nosed about just a couple slides. Zero in on your main theme around the human microbiome, because it's just to give you a quick snapshot of what that agency's interest is in that area. And then we're going to have a discussion between us about what each person might have called out as an obstacle. But the reason I asked them to sit in alphabetical order and ask the audience to move forward is I didn't want this stage, this table, to act as an obstacle to dialogue between the panelists and the audience. Okay? So I'm hoping to also invite conversation and debate and dialogue from the audience with the panelists. You've been talking about the obstacles for 3 days. They'll mention some. Let's see if we actually come to some consensus around the priority obstacles, or maybe it just ends up being a huge laundry list. We don't know yet. I'm also going to close, I'm going to forewarn you that I mentioned to everybody else, I'm going to close with at least one question I've been asked to ask, and that is if an agency or agencies were to mount a very large research cohort study, didn't have enough money to sample the entire microbiome, what would be the microbiome sample that should be biobanked for future analysis? I've got a lot of different answers from people I've asked around the audience, and I want to spend a little bit of time to get an answer from this group. So let's then get started. It's alphabetical order, and I've been asked by Lois. Where's Lois sitting? There she is. Lois Tully is from the National Institute for Nursing Research, and she's taking notes for the whole panel. So she's asking you that when you start to talk about your agency, just remind them which agency or office you're from when you're talking. Just say, and the VA says it thinks this, or FDA thinks this. That would help Lois keep track. Okay? Go ahead. Yeah. Oop. Walter's not happening. Oh no, Walter's not there.
Okay, let's see.
Okay, great.
Good afternoon. I'm Rajiv Agrawal, and I'm from Office of Research on Women's Health. And today I'm going to show you why sex as a biological variable is important for expanding knowledge and improving health of men and women. So the office was established in 1990 by the Congress, and in 1991 the office has this Women Health Initiative, and in 93 NIH mandated to include women and minority group in clinical research. Today, ORWH, or Office of Research on Women's Health, is the focal point for research on sex and gender influences, which benefits every human being, including like girls, women, boys, men. So the mission of the office is to examine sex and gender influences on health and diseases, strengthen and enhance research related to diseases, disorders, and conditions that affect women. I'm not going to the third one, which is like promoting for women in biomedical career. So what is the rationale for the new SABB or revised SABB policy? Over the years, there was over-reliance on male animals and cells, inattention to sex effects, lack of transparency, inconsistent reporting of sex-specific findings in publications, which led to incomplete knowledge base, risk of erroneous conclusions, irreproducible results, toxicity surprises, erosion of public trust, and of course, not a good return on investment. To give you an example, oops, what happened? So this is aggregated data, and it looks like that there is no treatment effect in that compared to the control group. Whereas if you disaggregate the data, which has been shown by McLeod, and they used PARP inhibitor PJ34, and when they treated the male animals at the time of ischemic onset, they saw reduced total infection in male mice compared to the saline-treated animals, whereas in the females, this has a significant increase in ischemic changes compared to the controls. So still the default biology in the clinic is 70 kilogram male, and in the lab predominantly male cells and male animals. Because of this reason, 8 out of 10 drugs withdrawn by the FDA during 1997 and 2010 had more adverse effects for women than men. So we all agree that science isn't science if it is not reproducible, as mentioned by Roth and Rox. So because of this reason, NIH clarified this policy and expects that sex as a biological variable will be factored into research design analysis and reporting in vertebrate animals and human studies. This is just to show you that sex differences in immunity results in disease bias, and on the left-hand side, the diseases are more prone to females, whereas on the right-hand side, on the male side. Since this is a microbiome meeting, it will be not good if I don't talk about SABV in microbiome. So this is a recent publication where the authors have shown that host responses to pathogen bacteria and beneficial microbes exhibit host sex specificity. They used Mycobacterium as pathogenic bacteria and Lactobacillus as probiotic and treated biopsy mice, males and females, and they analyzed the immune profile. And you can see very different immune profile in males and females. So this is a metagenomic analysis which shows difference in colon microbiota between males and females. Because of the time, I'm not going to go into the details, but you can see it's a very different microbiome analysis or revealed in males and females. And thank you so much.
Okay, well, I'll start before my slides are up there. So I'm coming at this from a slightly different standpoint. So we're a regulatory agency, obviously, from the FDA, but we also have a significant research program at the FDA. So what I want to go through right now is the research that's being done in terms of microbiome at FDA throughout our various centers. And I'm at the Center for Biologics, which I'll talk about later. Maybe? There we go. All right, so I broke this up into 2 offices. I have the Office of Foods and Veterinary Medicine, and then after that I'm going to talk about medical products and tobacco. But I also want to point out this logo that's going to pop up all over the place. This is the National Center for Toxicological Research. This is a center at FDA that is research only. They don't have to do the regulatory work that the rest of us do. But they're really there to support research into regulatory-based questions, and we all, I think every center, they're going to pop up everywhere. We all have collaborations with them, and I want to make sure that they get the credit they're due here. So just to go through the foods and veterinary medicine side, we have the Center for Veterinary Medicine. They're really focusing on projects looking at antimicrobial pressure on microbiome in farm animals and in ecosystems, and resistance genes and the potential of the microbiome to serve as a reservoir for antibiotic resistance genes in what is termed the farm to fork continuum. We're going to be eating these things, our microbiome is going to be exposed to these things too. And then the Center for Food Safety and Nutrition, I have listed down here. And CIFSAN, they're really doing a lot of research into the microbiome, and I've listed a few things here, and I'm generalizing. There are a lot of projects involved in these different things, but the role of diet and nutrition in the microbiome, specifically also looking at the microbiome of foods, what organisms are present in the foods. These are things we're going to be ingesting, kind of goes along with diet. And then pathogen detection in foods in the context of that microbiome. The Office of Medical Products and Tobacco, I'm going to talk about three different centers. The first is the Center for Drug Evaluation and Research, and the projects that I have listed up here are looking at the effects of drugs, in this case antimicrobials or TNF agonists, on the microbiome. And so we have a few research projects going on in the Center for Drugs and also in collaboration with NCTR, as I said before. The Center for Tobacco Products has research ongoing looking at smokeless tobacco and the oral microbiome. And there are a few projects based on these, and there's a publication listed here from some in vitro work that was done, and again in collaboration with NCTR. Finally, the Center for Biologics is the center that I'm in, and some of this is my data, but if you think about it in terms of products, we're focused on fecal transplants, live biotherapeutic products, which some of you will call probiotics, and also phage therapy. And these are the things that we are regulating in terms of microbiome-based products. So we have projects looking at FMT. This is data on C. diff, and also projects on LBPs, looking for mechanisms or ways to measure purity and potency in these products that are full of bacteria intentionally. And hopefully you saw some of the posters that we had out today. And again, these are also in collaboration with NCTR. And the last thing I want to mention is Hive. Hive is based in Sieber, but supports researchers across the FDA and also regulators. Hive is our bioinformatics core, and they've done a really good job of starting to incorporate some of the commonly used microbiome analysis tools into their systems, both on the research side and also a separate system for analyzing regulatory data that come in that are microbiome specific. And I just wanted to acknowledge the people who contributed data slides and other information for this. And I'd say, just to go back, I don't have the gaps, potential gaps listed up here, but not so much from the research side, maybe thinking about this in terms of the regulatory side. I think some of the gaps would be moving beyond association and actually starting to get into mechanism, and I think a lot of people have said that today. But also assay validation. You've got a lot of steps in the process of looking at a microbiome and to really make sure those are being consistent across the board in order to have regulatory quality data.
Okay, mine is going to be really short, and I don't have any data to show, but I am in the Office of AIDS Research, and this is an office that's in the Office of the Director for NIH. We're responsible for the scientific, budgetary, legislative, and policy elements of the, So we're responsible for the scientific, budgetary, legislative, and policy elements for the NIH AIDS research portfolio here at NIH. We've had a longstanding interest actually in the microbiome, probably close to when the HMP was being developed, so around 2007, NCI actually had a workshop which focused on HIV, cancer, and the microbiome. And then in 2009, we added microbiome as a specific area within our Trans NIH strategic plan for HIV research. And we hope that this would encourage not only the institutes to fund more research in this area, but also as a call to investigators in the HIV field to submit applications in this area. And we really didn't see sort of our investment in the area increase until probably about 2012. That's when we can see some appreciable research support being specific for the microbiome. And as you can see, within a 5-year period, the investment has just about tripled, and I suspect that in 2017 it's probably going to be substantially more. So in the Office of AIDS Research, as I mentioned, we have these specific areas or priority areas that focus on the scientific priorities for HIV/AIDS research. And within those priority areas, one being reducing incidence, therapeutics, comorbidities, co-infections, basic research, as well as research toward a cure, you can see how the investment has been broken out. And if you can look at 2017, which is our most current data, or 2016, I think, not 2017, most of the research is being done looking at basic aspects of the microbiome and HIV, as well as areas to reduce the incidence of HIV in at-risk populations. Some of the research, especially in reducing incidence, I think was presented here, and there were quite a few posters looking at the vaginal microbiome and how it impacts HIV acquisition as well as treatment. And these are some of our high priority areas for HIV microbiome research here at NIH. Sitting here through the 3 days of the workshop, I can probably add about 10 or 20 more things here, but these are things that we have been working on as well as those that we would like to further invest in. It's a long list. I don't know if I should read it, but let's see. The impact of the microbiome on HIV risk, acquisition and disease progression, the role of the microbiome in HIV treatment effectiveness, including prevention interventions such as PrEP, which is pre-exposure prophylaxis. The microbiome is a biomarker for the measurement or the assessment of HIV disease and overall health of an individual. And actually, there were quite a few studies that were presented here. I think that will give us some clues as how to do that. How to influence and perturb the microbiome to improve HIV treatment, prevention, and pathogenesis. Something that I didn't hear much about was the geographic diversity and how it influences the establishment and composition of the microbiome. And for HIV specifically, how that impacts HIV transmission, establishment of infection, and the outcomes of disease. And last but not least, the impact of maternal microbiome and dysbiosis on the health of HIV infected and exposed uninfected infants and children. And while this isn't an exhaustive list, I think these are areas where we would like to see a lot more research on the microbiome field in HIV research. And I think that's it. Thank
you. Hi, I'm Linda Chrissy from the Office of Naval Research, but I'm here representing the Department of Defense, specifically our recently established Tri-Service Microbiome Consortium. The consortium is composed of both research sponsors within DoD of microbiome research, as well as performers at the various DoD labs in this space. So key purposes of our organization are to enhance collaboration, cooperation, communication of microbiome research among DoD organizations, serve as a forum for sharing of microbiome-related resources, et cetera, monitor global advances in this area and provide that information to our leadership, and then to engage our operational end users to identify specific areas of research that might benefit them. So in many ways, there's parallels to the organization that NIH has established, although I'd say we're probably a little more compact. So unlike the NIH, though, DoD's interests in microbiome, I would say, are complementary to the NIH, but definitely distinct, and probably distinct from many of the other agencies on the panel. We're not really focused on disease. We're focused on enhancing readiness and resilience of our warfighters. So we do, across the DoD, have investment or research going on in many different microbiome areas, including the ones I've listed here. So for the gut microbiome and gut-brain axis, the kinds of things that we think about are the kinds of stressors that our people are exposed to. Some of the environments they work in are really rather unique, and we need a better understanding of how these stressors affect the ability of those people to function effectively for long periods of time. So for example, since I work for the Navy, I'll share the submariner case. So we have people who are in a very artificial environment with no sunlight, with slightly elevated CO2. They're sharing bunks. They have an altered circadian rhythm cycle. You do have men and women on the submarines, which is another factor that we might need to consider. They eat the same food. They run out of fresh food very quickly. And so we are asking, how does the microbiome change, and are there signatures there that could help us understand perhaps some, how to make these people more resilient to living in that kind of very artificial environment. It's not unlike NASA and what they go through, although we don't have a representative here today. So Also, with the gut microbiome, we have interest in the Army and the Air Force in understanding biotransformation of toxic industrial chemicals that people might be exposed to. On the skin, we think about skin infections. So some of the boot camp training that personnel are exposed to, they're crawling around in the dirt a lot and often develop skin infections. Are there ways to predict that or prevent that? Where understanding the microbiome can inform those processes. Wound healing is another one. And then in the lung, also understanding exposure to toxic industrial chemicals and ways that the lung microbiome we might be able to understand that better and insulate our people from those effects. That was it. That advances the slides. And then here's a pointer.
Good afternoon. I'm here to represent Department of Veterans Affairs, Office of Research and Development. Our main goal at ORD is to do research in the areas which are of interest to veterans. We have a large number of veteran population in our hospitals, so we are more veteran-centric, and veteran-specific research we are doing these days. And I have provided some of the details in slides, which are, I think we are not showing here, but in the deck. I'm focusing here only those areas which are more focused in the microbiome specific. The ORD supports microbiome research related to improving the health and care of our nation's veterans. It supports microbiome research our research spans a spectrum of diseases and medical conditions, including investigating the role of microbiomes in alcoholic liver disease and cirrhosis, characterizing components of the nasal microbiome that could prevent transmission of methicillin-resistant Staphylococcus aureus MRSA in VA hospitals, and determining the role of the microbiome in preventing or reducing infection at the implant sites of veterans receiving osteointegrated prosthetic lower limb implants. The majority of VHORD supported microbiome research is investigator-initiated and based at VA medical centers across the country. The breadth and scope of microbiome research has increased steadily over the last decade as more advanced tools in next-generation sequencing and bioinformatics have become more widely available to the VA researchers and academic affiliates. The magnitude of microbiome research within the VA is anticipated to continue to increase in the coming years as more VA investigators gain expertise in molecular and genomic science methods and bioinformatics data analysis techniques through their ongoing training and research activities. Currently, we are trying to further explore whether changes in microbiomes play in the high priority areas such as PTSD, TBI recovery, addiction, and suicidality or responses to therapy, especially in Gulf War veterans. Related areas such as one of these is like inflammatory irritable bowel syndrome. Thank you.
Hi, everyone. Excuse me. I'm Scott Jackson from NIST. NIST is the National Institute of Standards and Technology. I'm going to talk to you about some work that we're starting over the last few years related to microbiome. So just an example of how NIST has played a role in this field previously. A couple years ago, we released a human genome reference material. It's the most well-characterized genome in existence. It's been sequenced to thousands of X coverage by all different sequencing platforms. And it's used by industry to assess the accuracy of DNA, of human genetic testing. So just an example of how NIST has played a role in the genomic space recently. We are in the Department of Commerce, so industry is our stakeholder, but we typically interact with the biotechnology industry that is regulated by the FDA, so we have a close working relationship with the FDA and our industry stakeholders. And typically when we collaborate, it's to develop industry standards like the Human Genomic DNA Reference Material, an industry standard. So we have close ties with FDA as well as our industry stakeholder. And I should mention that we're not a regulatory agency. So what are our ongoing microbial efforts at NIST? As I said, our program is relatively new. It started around 2014, 2015. So we are in the process of developing microbial reference materials, both genomic DNA, whole cell, mixtures, as you might imagine, that are needed for the microbiome community. We are hosting interlab studies, or challenges as we call them. We actually just finished one up and began looking at some preliminary data from it and found some very interesting findings just from a quick and dirty interlab study that we hosted over a period of about 6 months. We host annual workshops. We alternate years between the focus on standards for microbiome measurements and standards for pathogen detection. Actually, this week, Monday, Tuesday, we had our semi-annual standards for pathogen detection workshop at NIST in Gaithersburg. It was a great workshop, I thought. We had the world leaders come out. I want to point out that standards for pathogen detection, these are the clinicians, the infectious disease clinicians that are looking for pathogens. But that community is beginning to use metagenomic methods just as the microbiome community is. So I sort of jokingly tell our audience at these meetings, we have the same discussion every year, whether we're talking about microbiome or whether we're talking about pathogen detection. It's the same needs, it's the same measurements, it's the same underlying problems. So next year, 2018, will be our Standards for Microbiome Measurements workshop. Keep an eye out for an announcement on that. We host consortia. I encourage you all to visit the website, which I didn't put on here, but it's rather easy to remember, microbialstandards.org. So it is a one-stop shop for everything microbial standards, including the home of our new consortia, IMSA, which is the International Microbiome and Metagenomic Standards Alliance. We have monthly WebExes. Actually, I see I did put the website on there. It's open for everyone. It's an open gathering discussion forum for talking about anything microbiome standards related, whether it's sample collection and storage methods, whether it's DNA extraction techniques, etc. We have internal laboratory research programs that are focused on developing new measurements. I'll probably talk about that a little bit later when Lita pings us about gaps. And as Lita pointed out, we're represented on interagency working groups and other consortia like the Microbiome Coalition. So with that, I will stop. Oh, sorry.
So my name is Cliff McDonald. I'm here representing the CDC, the Centers for Disease Control and Prevention. I'm in the Division of Healthcare Quality Promotion. We used at one time, we're called the Hospital Infections Program. That's our focus, including Clostridium difficile, the major microbiome-mediated disease right now and treated disease. And I'm here to talk about antibiotic resistance in the microbiome. It's where we think public health and the microbiome are meeting right now in current time. And we talk about this in terms of a forest. That a healthy forest keeps most of these multi-drug resistant organisms out or off the skin or out of the nares or out of the gut. That microbiome disruption, usually from antibiotics, is like a forest fire through the forest, the ecology, providing an ecological niche for these organisms seen here, methicillin-resistant Staph aureus in the nares, carbapenem-resistant Enterobacteriaceae, abbreviated CRE, what we call our nightmare bacteria in terms of resistance. Clostridium difficile, what we've called the deadly diarrhea. It kills 15,000 to 25,000 people a year in the United States, close to 500,000 infections. These organisms take advantage of this disrupted microbiome and take root and proliferate. And there they spread to other patients. So our work is around developing microbiome solutions to the antibiotic resistance problem, trying to predict the impact of different antibiotics on the microbiome, tailoring antibiotic use. One of the major efforts to prevent antibiotic resistance is to better use those antibiotics. We call that antibiotic stewardship. And in the future, it's likely that microbiome diagnostics will play a role in that. Develop and test microbiome diagnostics and the protocols, and even development of some of those tests. And then also where we can, working with other agencies, FDA, NIH, NIST, and others at this table, support the development of therapeutics that could restore or protect the microbiome. We have currently 3 collaboratives discovering new ways to protect patients. We have 4 prevention epicenter projects. These are centers of excellence, 11 of them across the United States, and several of them are doing microbiome projects. And we've funded over 15 studies and contracts last in 2016. It'll certainly be much less than that in 2017. But these are mostly around looking at predicting microbiome predictors of becoming colonized or once becoming colonized, dominated, or infected. This is poor George. He's often a patient who becomes colonized or infected with a multi-drug resistant organism, and we've used him in previous public communications about this problem. And in this case, from the far left, he has a normal microbiome. He gets an antibiotic, and that's disrupted, and then a multi-drug resistant organism or a genetic resistance element comes into him and then develops colonization, followed by dominance and transmission and infection. And so we are envisioning the development of microbiome indices. There are a lot of the things we've talked about here. We've heard about dominance. We've heard about keystone species, protective species. All these are things that we want to try to catalog and categorize and develop into either into diagnostics, but also to use as yardsticks for new drug development, antibiotics, and then also the microbiome protectants and restoratives. I think that's it. Yeah.
Good afternoon. My name's Jack Okamuro. I'm from the USDA Agricultural Research Service. I'm in the Division of Crop Production and Protection. We have 2 agencies which are the primary research agencies for the USDA. Agricultural Research Service, or ARS, is the internal intramural research agency, and National Institute of Food and Agriculture, or NIFA, is the extramural grant awarding agency. Before I get into much about agriculture and relating that to the human microbiome, how many of you are going to, after this meeting is done, are going to take off and go chase the solar eclipse? Anyone here? Just a few hands. Okay, well, a few years ago, you could have described the microbiome as the dark matter of the universe of life. But it's clear from this symposium that that's rapidly changing. And you guys are the Hubble telescope for the microbiome, okay? So keep up the good work, and also think about agriculture, all right? Let's take a few steps out of the laboratory now and into the field and talk about what the potentially high impact microbiome research targets are at the USDA. And they cover both human nutrition and health, but also veterinary and agricultural production. One of those high impact targets is to prolong the effectiveness of antibiotics for treating people, as well as animals, and also crops. How many of you said crops? Anybody? Okay. Yeah, Jim does. Thank you, Jim. How many of you have heard about citrus greening? Honglong Bing. Just 5 years ago in Florida, so this is a disease of citrus. It's been devastating in Florida. Over the last 5 years, Florida State has gone from producing 270 million crates of citrus for juice to just 70 million currently. So the industry is just hanging on by its fingernails. The disease is caused by a bacterium called Liberibacter asiaticus, and right now it's not under control. And we, growers and research scientists, are trying a number of strategies to control the disease, and of course one of those is to use antibiotics. Okay. Antibiotics are a tool that we use not only on the animal health side and the human health side, but also on the crop side. So we're still in the middle of this war, and we don't know how it's going to end up. But antibiotic resistance is also a concern on the agricultural side. Okay, give you another example of potentially high-impact targets. How many of you had a sandwich today for lunch? Okay, made of wheat. Now we're going to talk about some of the good guys in the field, and we'll talk about soil health. And of course, there are good microorganisms in the soil which are very beneficial. If you go to the Palouse of Washington State, you take naive land and try and plant wheat seeds, which Washington State is a big wheat-producing state, chances are your field will come down with something which is called take-all disease. Take-all disease. Basically, it wipes out that field. Very high probability. And if you get a severe enough case, if you come back the next year and plant your wheat, it may do better. And over several years, if you keep planting wheat, it'll do better and better until now you have a wheat-growing field. So there is a species of, I didn't tell you what was causing the disease, but I'll tell you that because I can't pronounce the Latin name. I'm terrible with Latin names. But there are Pseudomonas species who produce antimicrobial compounds which are active against the disease-causing organism. So they're good players in the microbiome as well, and we're just as interested in those in agriculture as we are in human and animal health. Anyway, I'm going to be running out of time here. So what are the key gaps? We frequently have stakeholder listening sessions, not only here in the urban centers, but also in the agricultural producing regions. And one theme that we hear all the time is workforce. So not enough computational biologists, scientists who can speak the language of genomics and genetics, but also agronomics and production and plant biology and plant pathology, animal pathology, animal health, as well as human nutrition and human safety. So I'll stop there. That's my contribution for key gaps.
I do need to clip it. Thanks. Good afternoon, everybody. My name is Jim Olds, and I head up life sciences at the National Science Foundation. And Jack's agency and my agency actually do a lot of work together. We also work with our friends at the NIH. But I'm a molecular neuroscientist trained at the NIH originally, but I've become fascinated with plant biology, and it's because food production is so incredibly important. Our survival depends on food production. So, Jack, I'd like to echo scientifically where you're coming from. I think it's incredibly important. Look, the drivers of microbiome strategy come out of the White House Life Sciences Subcommittee on National Science and Technology Council, which I co-chair, and they chartered the Fast Track Action Committee on Mapping the Microbiome. The report of that group found less activity than expected in agricultural microbiome research, particularly in food-based studies, viral microbiome research, and applied microbiome research and tool development compared to basic microbiome research. So some activities that are ongoing at our agency, which has a budget of about $8 billion a year, Microbiome research in life sciences has flourished by letting the research committee drive the important questions. This has resulted in a substantial investment in microbiome research that is part and parcel to core research across all of the Biological Sciences Directorate, which I head. This approach allows us to strategically focus on a few programs where we believe investment could have the greatest potential to impact broad research communities in practical ways. The microbiome portfolio at NSF has an important and direct and indirect impacts on human health and on the furtherance of microbiome research. These are a few examples of the activities. The Plant Biotic Interactions Program supports research on the processes that mediate beneficial and antagonistic interactions between plants and their viral, bacterial, oomycete, fungal, plant, and invertebrate symbionts, pathogens, and pests. So basically what we have here is a complex adaptive system going on that is absolutely critical to food production. All right? The program supports projects focused on current and emerging model and non-model systems in agriculturally relevant plants. Now, the ERICAPS network comprises 9 partners from 8 European countries and the USA, and 11 observer countries, 10 of them funding organizations, and is coordinated by the UK BBSRC, one of their research councils, our counterparts in the UK. ERICAPS initiative aims at deepening and enlarging European cooperation in the area of plant sciences, which should significantly help plant sciences to both address current and future challenges in food and non-food crop production. Think healthy, safe, sufficient food and plant-based products, chemicals, and energy. You know, one thing that I've learned coming to NSF is that your photovoltaic cell is about 20% efficient. The happiest plant in the happiest crop field in America is 2% efficient, maybe. So you could get depressed about that, but there's a lot of positive upside there also. And that's one way of thinking about where we could go in terms of improving food production. The EDGE program encourages the development of tools that can impact broad communities of investigators. Projects of interest could include, but are not limited to, elucidating fundamental principles by studying multiple microbiomes across different ecosystems and the development of computational and modeling tools for studying microbiomes. I'm going to jump ahead here and talk about our EID program, which we do in partnership with the NIH. That's the Ecology and Evolution of Infectious Diseases. So think Zika and think of hantavirus. So one focus of the program is to gain insights into the dynamics of infectious disease systems that require integration across several temporal, spatial, and functional scales, including molecular, individual, population, societal, and of course ecosystems levels. And I'm really excited about how that's going to intersect with our National Ecological Observatory Network, which we are now just finishing completion on. That's a $500 million approximately construction project, which has ecological sensors going from Barrow, Alaska to Puerto Rico, and actually stores archival soil samples and bycatch, which on a systematic basis actually can really contribute here. So here's a current and relevant example. I want you to think about this one as sort of, think the Human Microbiome Project and the Earth Microbiome Project. So the tools here in this project by Frank Stewart as the PI at Georgia Tech will provide improved methods to organize community gene expression data, that is metatranscriptomes, into metabolic pathways which inform predictions on how biochemical processes transform matter and energy. The developed software tools will be made available to the research community as standalone open source packages and deployed on common cloud computing environments. Thanks so much.
How do I make it go?
You hit the right button.
Oh, there it is. Oops. Thanks. So I'm Mike Sayre. I'm from the National Institute on Minority Health and Health Disparities.
NIMHD is the newest grant-making institute at the NIH. It was elevated from a center to an institute in 2010 with the Patient Protection and Affordable Care Act. Under our new director, Dr. Eliseo Perez-Stable, NIMHD has organized its extramural minority health and health disparities research efforts around 3 broad priority areas, and they're listed here. Integrative biological and behavioral sciences is my area. I'm the chief of that branch. We also have clinical and health services research and community health and population sciences. And I just want to take a moment here to say, when I was a graduate student in the 1980s, my work was with the bacteriophage that infected Bacillus subtilis. So I'm really glad to see phage back in vogue here as a potential therapeutic agent. That's very exciting. Each of these areas focuses on a range of health determinants and processes that contribute to poor health outcomes and health disparities in disadvantaged populations. And some examples are listed here, including human microbiome contributions to health and disease. With respect to microbiome research, NIMHD is a disease-agnostic institute, so we're interested in microbiota at all body sites, so that's going to make it hard to answer your question at the end. But we're especially interested in research on human-host microbiota interactions that may contribute to health and disease in disadvantaged populations, and how those interactions are influenced by behavioral, social, and environmental factors. So in the interest of time, I'm just going to briefly summarize 2 new examples, just 2, from our growing microbiome research portfolio. This project, led by Robert Kaplan, Rob Knight, and Robert Burke, examines the role of the gut microbiome in pre-diabetes and diabetes in Latinos. It leverages the NHLBI's Hispanic Community Health Survey, a study of Latinos, longitudinal cohort study. And this ancillary study will test the hypothesis that specific gut microbiome patterns are significantly associated with pre-diabetes and diabetes in this population. The project will collect and determine genetic composition of fecal microbiome from over 2,000 cohort members. And I think this is the first major epidemiologic project focusing on Hispanic Latino populations, their microbiome. The next project, the second project, led by Stephanie Engel at the UNC Chapel Hill in collaboration with Greg Buck at Virginia Commonwealth University, leverages data from a diverse cohort of women enrolled both in the NICHD's Pregnancy, Infection, and Nutrition, or PIN study, and the HMP Phase 2 multi-omic microbiome study, Pregnancy Initiative, or MOMS PI study, to investigate mechanisms by which unfavorable vaginal microbiome profiles may contribute to significant disparities in preterm birth among African American women. They'll be looking at the influence of maternal polymorphisms in innate immunity genes expressed in the vaginal mucosa, association between maternal psychosocial stress and depression, microbiota and preterm birth risk, influence of behavioral risk factors on vaginal microbiota, such as poor diet, smoking, and other health-related behaviors, and metagenomic analysis in a subset of preterm birth cases with infectious etiology, particularly bacterial vaginosis. So I know we're going to move quickly to the Q&A session, but I just want to encourage everyone to think about submitting your best ideas in microbiome research as it relates to minority health and health disparities. We're aggressively growing our portfolio of investigator-initiated research projects, so now's a good time to think about NIMHD. And I should also say that we are very interested in precision medicine. Last year we funded a major center grant to Stanford University, and Mike Snyder is the leader of one of the sub-projects on that grant. The PI is Mark Cullen. We're open for business and looking for your good ideas. Thanks.
So here's the experimental part. I think what I want to do is have one or more of you elaborate on the obstacles that you named very quickly in your intro, particularly as you heard the gaps, needs, and challenges also voiced by the audience? Like, what resonated with you when you were listening to the talks over the last 3 days? Or have you identified an obstacle that you hadn't heard yet? Anybody? For those of you who were here. Go ahead, Linda. Jump in. Yeah, state your name. Agency.
So, Linda Chrissy from the Department of Defense. So, I wasn't here for the first 2 days. I did appreciate the comments on gaps and challenges from the speakers today. One challenge, I'd say, that I see anyway as a funder of research for the Department of Defense in this area is when we go to our operational communities, they ask us, what are you going to do to help us? And for many of the areas that I'm interested in, we have animal studies, but there aren't any human studies. And yet it's very hard to contemplate, taking people out of a mission and doing research studies on them. So that's a question I have a hard time answering. What am I going to do for the operational folks with studies that are still at the animal research stage at best?
Could I ask someone to just turn on the house lights? There's no reason for them to be dimmed. Anybody who's in the booth in the back? Or
Can I jump in with the gap?
Please do, please do.
Who here in the audience in the last couple of weeks in Nature read the paper on an ecosystem on a leash, talking about microbiome? And it was really interesting because it was the concept of the fact that the microbiome, both in our gut and the microbiome that exists in the soil that interacts with crop plants, for example, is really itself an ecosystem that is in this very complicated relationship, obeying complicated rules of the road with either its host or the plants that it's nearby. And those rules of the road strike me to be a gap space, akin to the rules of the game of chess. But the rules of the road by which that massive ecosystem interacts with host strikes me as a big gap area that's worthy of all of our agencies focusing on.
I'd like to second that just from the perspective of Clostridium difficile. It's an organism we think of as a healthcare-associated pathogen, but we realize its really reservoir is probably in the community, and we really don't know where it diversifies out of the human host, and so it's just a perplexing question. And you could also bring it to when you don't have a soil around, and I think we heard that today. In healthcare, for example, we have microbiomes in healthcare that are separated from the soil, but they develop some characteristics of that interaction between the human and the soil. Now it's the human and the healthcare facility, the drains, the surfaces, and the people coming in and out.
So one possible approach to some of these areas is, one approach might be to try to develop some models. And I think at NIH, I don't see as much model building as I did when I was at NSF. So any thoughts, Jim, about what's happening in the model development area at NSF and what else is needed?
Well, models are really important to us, and not using the operational definition of model organisms. But models are really important to us. And basically, what that gets at is a theoretical framework. So gets us out of the stamp collecting and into the predictive science. We do support that. We're very enthusiastic about supporting that. And actually, what I would say is I think a lot of our sister agencies are interested in that also. I'll just note that another charter creation of the Department of Agriculture is the Foundation for Food and Agricultural Research, which I sit on ex officio on the board of, and is able to actually invest directly into basic research along those lines also. Sally Rocky, formerly of NIH, is the executive director of that organization. They have a bank account of $200 million, courtesy of the Farm Bill of 2013. So I think there's a lot of potential in this area, and I'd like to see some creative ideas come out.
So I think for HIV research, some of the gaps that I see, one is, again, the animal models. So we do a lot of our research in non-human primates because that's the best model for HIV. However, there are other small animal models that we could explore because non-human primates are quite expensive. Another gap, I think, is sort of the multidisciplinary research. We have a lot of HIV expertise. We don't have a lot of microbiome expertise, and so I think trying to link the two communities is something that NIH should be promoting. And I think maybe the third gap would be, just went out of my head, sorry. Oh, it'll come back to me, sorry.
Scott Jackson from NIST. I wasn't able to attend the first day of this conference, but I'm sure Lita mentioned in 2015, the White House Office of Science and Technology Policy hosted the Fast Track Action Committee on mapping the microbiome. That committee was tasked with identifying gaps, impediments, et cetera, and one of the unanimous gaps that was identified by that committee was the need for standards for microbiome, reference materials, reference data, et cetera. So NIST has perked their ears up. We have workshops to bring in our stakeholder community and say, what does that mean? A thought exercise that I like to do when I have people gathered like this is ask, in your mind, what is a standard for a microbiome measurement? What does that look like? And there's no right or wrong answer. Some people say, well, it's a mock community. Some people say, well, it's a procedure, it's a protocol, maybe it's data, and these things are all correct. So NIST has been thinking about this and working towards delivering stakeholder community needs, both in the diagnostic side as well as the therapeutic side. So one of the things I'm most passionate and interested about is this emerging field of live microbial biotherapeutics, because these development of these next generation therapeutics are held to the most stringent regulatory levels. So measurement science is critical in that field, demonstrating purity, stability, identity, viability, et cetera, of your new live microbial therapeutic. These are measurement challenges. So NIST traditionally develops new measurements in addition to developing standards. So that's something we think a lot about. And on the other side, NIST has lots of engineering and physics expertise, and we typically build new devices, new instruments, and we have started a new research program that is building in vitro tools to model and understand microbial community resilience. So microfluidic devices that allow us to inoculate small numbers of bacteria and then watch how they equilibrate, how they become a community, how they stabilize. I call it Le Chatelier's principle in dimensions, right? So we're just beginning this, but we have a lot of microfluidic engineering expertise that we're leveraging to begin to understand how microbial communities become robust or unstable or stable.
But Scott, in those kinds of research activities, though, because you're primarily an intramural organization, you don't give out grants, or how do you partner with academics and medical researchers?
So we often partner with industry through CRADAs. We partner with FDA. A lot of our biological standards that have been developed, like our human genome in a bottle, have been funded by the FDA Center for Devices. So they need standards to help them regulate industries, typically diagnostics and therapeutics, so they fund us to develop those standards. Academia is typically an informal collaboration. Actually, the group that developed the human genomic reference material, Mark Salet's group, they actually moved their lab from NIST Gaithersburg to Stanford University. So they actually have a NIST lab on Stanford campus. And it was specifically done to allow young researchers to engage with NIST early on so that throughout their career they understand what NIST does, metrology, et cetera, and it's been very successful so far.
So I have a question too. Okay. So I have heard a lot of from investigators who are in academia They say it is very difficult to get standards from NIST because they are very expensive. And our budget does not permit that much money for getting those standards. They want to use them, but the cost is a main factor. So how do you deal with that?
So typically, our standards are developed for industry, and industry usually doesn't have those financial challenges, I guess. As far as the price goes, I can just tell you that it may take us 10 years to develop a standard, a reference material. But the price is based on how much it costs to manufacture that material. So for example, when we're making a mixed pathogen DNA reference material right now, it's costing us under $100,000 to make it, but we'll spend the next 5 years characterizing it, right? So the price of the material is based on how much it was cost to manufacture, not the R&D that goes into the characterization of the material. That's sort of the model that NIST has used on pricing their reference materials. Yes, we have some reference materials that are $50,000 for a single unit and some that are obviously much cheaper than that. So any academics out there who have the issue of needing a NIST reference material they can't get their hands on, contact a researcher at NIST. If it's me, I'll mail you the material. I didn't say that out loud. But we want people to utilize our materials, and we don't want to impede research because you can't afford it. That's the last thing we want to do. So, yeah. So contact a researcher at NIST, and if you need something, we'll partner with that researcher and collaborate. And, I need help characterizing the reference material that we're making. So what we're doing is we're selectively giving that reference. It's not a reference material, it's a research material until we stamp it reference material. So we're sending that research material out to collaborators and saying, if you sequence it, share your data with us. And then, that's a crowdsourcing way of helping us build confidence in what the material is. It's an informal interlab study is what it is. So it helps us build confidence in our reference materials.
I want to ask something. I don't know if Jen Vetweiss is in the house still. I know Aaron Marcus is still in the house. Could you just take a moment and let us know if anything has come in through our live tweet? Or, here's Jen, okay. Or Gmail. Any questions for the panel? Oh, okay.
It's interesting. If anyone is still on Twitter and is listening, I have the feed in front of me, and I'm happy to pick up a question.
Great. I just want to make sure. Thank you. I think Stacy remembered her third thing.
Actually, it was as soon as Scott started talking because it was on standards. So standard methods, standard ways of doing research in HIV and the microbiome, I think is an important area that we need to focus on as well.
So Jack Okamura here again. I just want to echo Jim's suggestion for models. So if we think where we want to be 5-10 years from now in terms of better being able to ensure human health, safety, the safety and security of our agriculture, models for, where are the sources, the reservoirs of the pathogens, the in the wild or in our agricultural systems, and how do human activities, how do natural activities affect those microbiomes and affect the structures of the communities so that we can better manage the risk and not worry about the next outbreak of Zika or Ebola or some of these other things. So I think models are very important.
Go ahead.
So I was really struck by the theme that came out through many of the talks about the need for more understanding of functional interactions between microbial communities and with the host, and the human host in particular. So from the perspective of minority health and health disparities, the human research is going to be critical. And one thing I would see as a gap for us is the underrepresentation of minority populations in human microbiome research, and this has always been a problem with omics research in general, but we really would like to see a greater representation of minority populations because we know there are some differences in microbiota that map to, continental ancestry, for example. So epidemiologic studies, mechanistic studies, patient-oriented studies, all of it, we would like to encourage recruitment of minorities.
Just as an observation, maybe I'm talking out of school here, but when the HMP had a fairly small cohort, and the healthy cohort studied 300 people, but I think 10% or so were Hispanic Americans, and maybe another 5 or 10% were African Americans. But there was a very clear metagenomic signal in that population, but there was a lot of pushback from NIH in general about highlighting and reporting those data. There was a suggestion that maybe we didn't have enough population size to make these declarations, but the scientific data was very clear. So part of what's happening is, I think, and NIH might be giving out two messages. Do these studies, but you have to be super careful because I presumed it was political sensitivities. So I just want to make that comment as an NIH person talking to another NIH person. So I know not everybody at the table here is a funding agency, and I did promise you we weren't going to talk about money, but is there any, are there major obstacles to trying to develop, I mean, Jim gave some wonderful joint agency activities, right? Like the Ecology of Infectious Disease has been around for a long time, extremely successful. Are there major obstacles to building more joint agency activities, or is that in fact the way to go to span this very large field?
So I think it is the way to go. And I think there are plenty of examples. I look at the partnerships that have come out of the BRAIN project between multiple agencies. We just had a meeting yesterday here on campus, the multi-council working group. And the synergies have really been incredible. And I think we can do that in microbiome also. And I think it's also a way to catch the imagination of the American public, to get different agencies with different stakeholders working together. So I think that can be very powerful. But most importantly, I think we have an obligation to leverage our finite funds to move the scientific needle as effectively as possible. And the way to do that is to work together.
I want to give the audience an opportunity to ask any questions of the panel. Again, this was not meant to be just a discussion amongst panelists. So as we continue our conversation, please stand up and ask your question. Jen.
This is a question from Natasha Shelby. She would like to ask, why are there so many experimental diets labeled high fat instead of high processing or low fiber? It may seem like a matter of semantics. It affects our interpretations, media fallout, and human action. There's a great body of research showing that high-fat diets can result in highly beneficial health outcomes. A ketogenic diet treating epilepsy is one specific example that has been well publicized. What if the results we see in these so-called high-fat diets are actually the result of high-processing low fiber and are not the content of fat? Is there value in being more careful with how we label these treatments and interpret these results?
Well, I'm not sure it's necessarily a question for the panel, so maybe I'll just respond to it quickly. I think there are other studies looking at other dietary components of our normal diet. High fat seems to be one of the more common ones. So whoever asked the question, I think you can take heart that there are many other kinds of diet and microbiome studies. Mike.
Yeah, listening to you guys talk, it's kind of cool seeing all the, common interests across these various agencies and things. It also seems like it's an amazing opportunity to launch projects that maybe wouldn't be so easy. And one I think of that I'm involved in, some of you may know this NASA project where they're profiling two twins, one in space, one on the ground. And of course, the space station is a very controlled environment. And so listening to the submarine environment, that's a pretty cool opportunity to be a very closed environment, presumably some, not open-ended in the nutrition side of things and the food side. So it does seem, to the extent that we could take more advantage of those situations, that would be a cool thing to do. And I don't know, do you guys actively seek that out, or how does that work in these situations? Probably for each of these agencies, there must be some niche that's very special that makes a very interesting experimental situation that we should try to capture. Thoughts? I used the submarine one because you launched that and it was a great idea, I thought.
Yeah, we probably won't be able to do that, but we do think of doing So, for example, having a research study on a submarine during a mission is not going to happen for obvious reasons. But we do think about exposure chambers where you can control different parameters of that environment and actually sort out those different stressors. They're very expensive studies, and we're sort of at the front end of trying to figure out what we might want to do in that space. And it would probably be a cross-service effort because the different agencies, for example, we may be interested in high oxygen or elevated CO2, and another agency, another part of the Department of Defense might be interested in low oxygen. So we'll probably formulate a suite of studies that could look at these things in controlled environments.
Sure, no, that would be terrific. And FYI, one of the things we discovered is that people's blood oxygen drops on airplanes. And so, that's a very interesting opportunity as well, where you might have a whole bunch of people, I don't see the Air Force represented here, but again, another very interesting, oh, you represent that one too, cool. So anyway, again, there's some very interesting situations, experimental situations that could be, and they actually have some implications, as you well know, for the general population as well. So.
Yeah, so throughout the meeting, we touched on topics regarding the ethics of these microbiome-based therapeutics. And you mentioned, the need to sample underrepresented groups and populations in some of these studies. I'm wondering if in the budget or the planning of these projects, you are trying to incorporate the potential ethical impacts of some of these technologies that are being developed.
Would you mind elaborating? I'm afraid I'm not catching
Yeah,
the
so
question.
for instance, let's say in FMT, you have a stool collections that are coming from all these people, and then you have people who are doing it at home because we have a technology that is being developed and is being proven to be very efficient. It seems that there's no infrastructure in the research to quickly implement it or make sure it is regulated in an easy way. So I don't know if with the projects that are being funded, you're kind of foreseeing the potential impact of some of these things that are being produced.
So I'm gonna stop you there just for a minute. I have nothing to say about funding. We don't fund, but FMT is being regulated. And we are doing a lot of things. We see a lot of FMT files coming in. So I heard it once before, and you just said it now, that FMT is not being regulated, and that is not true in terms of C. diff and in terms of many other applications. For C. diff, we do have the policy of enforcement discretion, but that doesn't mean that people aren't doing studies under IND for that indication as well.
If I could say more about that too, that is an enforcement discretion for multiply recurrent C. diff only, in which case the patient should be consented and it should be explained to them that it's experimental. In any other use of FMT, it is a drug, and an IND should be obtained. In fact, I know we've funded a couple projects where an IND is obtained to do a study specifically with FMT. So, but I think the question at large is still, a good one. That, in the general sense, I think you're trying to say that these things are available. It's a little different from that sense from a drug. Most other drugs, you can't make up in your kitchen, or you can't go to the bathroom and produce it, right? So, yeah. and then also just the very fact that there's microbes all around, and so. That touches on IP issues. It also touches on regulation issues, and it's probably beyond the scope of this meeting, I think. But I know that NIH has funded, and I don't know which group, I can't remember, but someone else can speak up to it, but has funded some look at the ethical, or not the regulatory framework of probiotics, for example, and FMT, and there's others in the audience who could tell us.
I think it might be. So there is an ethical, legal, societal implications program in my institute, National Human Genome Research Institute, and they did the regulatory framework of probiotics, funded that work. And I know there are groups within the LC, they call it LC, the LC community that are studying those very topics. It may be also being funded by other institutes like NIAID, but I'm aware of several investigators within the LC program in my institute. Mike.
So we funded 5 centers last year focused on various aspects of precision medicine. One of them I mentioned at Stanford. All of them, one of the priorities of the initiative was to look at the ethics and acceptability and feasibility of implementing precision medicine approaches in disadvantaged populations, minority populations. So they're all looking at that in various ways. That is a very important point.
Thanks.
Yes. Li Pingzhao, I have a comment. I think the biggest challenge or the gap is actually find out what a healthy microbiome should look like. And we know there's, for chronic diseases related with dysbiotic gut microbiota, it's not a gain of function like a chronic, like infectious disease. It's actually a loss of function. So you have lost the protective, healthy gut microbiota so that you may have some pathogenic or detrimental bacteria overgrowing and you lead to a dysbiotic, and that may be driving various chronic diseases or contributing to. So we should probably change, shifting from disease-centric to healthy-centric. So look at how from the very beginning of life, at various stage, how a healthy microbiota should look like, and what is the normal variation of the structure among clinically defined healthy people, and then using that as a standard or reference, and then compare that with diseased populations and identify targets.
So NSF's SBE Directorate, Social Behavior and Economic Sciences, is very interested in that. So our perspective on a lot of these issues is understanding the healthy. And that's quite distinct from the disease perspective, but I think it's very complementary to what goes on at NIH. And it certainly falls into the notion of what we see as our wheelhouse in terms of understanding basic mechanisms.
Thank you very much. Another very quick comment is the whole field probably need to bring more microbiologists and particularly microbial ecologists into the microbiome research. And I see that can benefit the human microbiome research a lot. Thank you.
2 questions. So one is that a lot of individuals showed that a big gap is being able to bring a lot of this microbiome research to humans. And part of that problem, I mean, there are lots of challenges to that, but part of that problem is that the probiotic I may use may be different from what another lab would use, which will be different from another lab. Although we've relegated probiotics to the realm of supplements, is it time to change that and make that more standard so that we can actually get some answers? That's number one. Number 2 is that another gap that people brought up is showing mechanistic relationships. And a lot of individuals who spoke are showing that through engineered bacteria, in which case the big question is, how will that finally translate into humans? Is there a plan for, like, what does an engineered bacteria therapy look like in humans in terms of what regulations would it have to go through?
So I can take the second one. I think it's going to depend on what the organism is and what the modification is, but everything is going to be reviewed based on the science. There's no directive that says, no, we can't have genetically modified organisms or anything like that. So there might be a little more work that has to be done depending on what the modifications are, but I don't see any major barriers to getting something through.
And those are not really, I mean, the IL-10 producing Lactococcus lactis has been around a long time, and so some of these issues have been addressed, I think, initially there. But
I can take a stab at the first question, too, regarding regulation of probiotics. So one of the things that I want to point out just for some people it's not clear. As you mentioned, probiotics are food supplements that are regulated by the Center for Food Safety, FDA Center for Food Safety, as opposed to these emerging live microbial biotherapeutics like SEER-109 that Seres Therapeutics is developing, which is regulated at a much higher stringency by the Center for Biologics that Paul represents. One of the things that CIFS, so in a previous life before I joined NIST, I worked for CIFSAN for 11 years. One of the things that we started looking at around 2014, 2013 is looking at probiotics more closely, at their identity and their viability, et cetera. And what we found is that oftentimes they weren't what they were labeled. So 10 to the 10th active cultures was sometimes 10 to the 6th active cultures, and sometimes there weren't the species that were labeled on the sides of the tube. But because there are no claims behind probiotics, they can't have any medical claims, then as long as they are safe, that's really the only regulatory criteria. Paul's looking sideways at me.
So the one thing I'll add, though, is that if you're using an off-the-shelf probiotic to treat a disease indication, if it's being used as a drug, that still has to be done under IND. Just because it's available at Whole Foods and it's out there as a supplement does not mean that you can just go ahead and use it in a trial against C. diff or against something else. So if it's used to treat, prevent, control, mitigate disease in humans, then it is considered a drug. So it does still have to come through and go under IND.
Okay. I guess I was referring to just daily use of probiotics. Like I take a pill every morning. I don't expect it to cure anything that's wrong with me. It's just a supplement. The last thing I'll say about that, U.S. Pharmacopoeia, which we all know has developed standards for the pharmaceutical industry, has recently started an expert panel on probiotics, and they are writing new guidances to the probiotic industry on the development of probiotic standards. So how do you demonstrate the purity, the identity, the stability of your probiotic? Stability obviously being viability. These things were really unwritten previously, and I know when these new biopharmaceutical companies like Seres came into existence, I've heard from some of them that they looked towards the probiotic industry for these standards. How did you guys ensure your manufacturing processes? And things didn't really exist, at least not in a well-defined way. So USP is developing new standards for the manufacturing of probiotics, and I know to a higher regulatory level, CBER is as well.
At a much more mundane level, standards, of course, are extremely important, but in clinical research, we can't even get blood pressure measured the same way in two different sites, as Owen White pointed out in his presentation. So we got a lot of work to do. The clinical research community has to really, in order to take this into humans, has to get real disciplined about measuring all kinds of things.
There is this Genome Standards Consortium, which Owen White mentioned on Wednesday morning, and that's a problem across all fields, where we're not collecting this. We're not agreeing on the same term for common properties. So if you want to do comparative studies, you can't do it because we're not using the same terminology for a particular property. That's true for ocean studies, that's true for soil studies, that's true for human studies. I chair a group called it's the group that helped build this workshop, the Trans-NIH Microbiome Working Group, and I want to start a dialogue with them about encouraging their PIs and putting in their RFAs a request, maybe not a requirement, but a request, that they include a minimum set of metadata with these common standards, and maybe that'll help start to build the ethic of utilizing common terms across common studies.
I wonder if we could get NCATS to partner on that through their CTSA
Yeah, let's do that. I'll find a way to get all that information out. Thank you. And I guess Rob was in line. Yeah.
So Rob Britton, Baylor College of Medicine. So at HMP2 kickoff, I made a plug for genetics, and Jeff Gordon so eloquently did it yesterday too. And I think the comment really is metagenomics, all these really neat associations, these new genes that we're finding. To get to the mechanism, you're going to need genetics. It's almost like microarrays 20 years ago that people were writing all these papers about microarray and expression data and making all these conclusions, and finding out later when they actually did the follow-up experiments, they were wrong. And I feel like we really are going to need to have a community-based effort, not just people like me who develop it for my one bug, but really identify the 20, 40, 50 organisms that we want to develop the genetic tools. And there's enough talent in this room, in this field, that if we had a sort of a concerted effort, I think we could build the tools that would really help us move from, really interesting and neat associations from all this technology we build around sequencing and get us to the function, because that's what you all want to put it in people, but we don't necessarily need to know the function of every drug we take, but certainly that will help us, I think, get to answers in the microbiome field, because these are living entities, not chemicals. So, thanks.
And Jack, in the agronomic field, with looking now trying to rediscover what the microbiome is of crop plants, that same argument must be made in your community as well.
Yeah, right. And the advantage of working agriculturally is we can do the experiment over and over again and change the variables. So definitely the advantage of using genetics, of using different germplasm, of different environments, of manipulating cultures and so on. It's very powerful. So think about animal and crop systems.
So Matt Henn, Series Therapeutics. I guess I have more of a comment than a question. And something I've been really struck by over the past 3 days and listening to people explain gaps in this conversation example, I've realized there's a bigger gap here that I haven't heard at all brought up, which is how we actually work together. And what I mean by that is that the gaps that we're hearing about, we've actually been hearing about for years. I've been on the academic side for the majority of my career. I helped launch Ceres about 5 years ago. So I've been on the industry side now for a handful of years. And I can tell you that all these things that we've heard over the past several days are gaps that just keep coming up. And I actually think the reason we're not solving these is for a couple of reasons. Number one, I think we have to get good government, academic, and industry partnerships going, because these are huge problems that require a large amount of money that no entity on its own can actually support independently. But to make that successful, we need to stop talking about what the gaps are, but actually define some attainable milestones and steps to move the progress forward that we can benchmark ourselves against. Because that's what I'm never hearing in any of these conversations, is sort of what are the specific milestones we want to achieve and accomplish as we move the ball forward. And so I guess I'd put a challenge out to this whole group to say, can we get a forum together? Can we get some working groups together that can actually start to define very tangible attempts or milestones that we want to achieve? Because I think that's how you're going to get alignment of everybody, and that's also how you're going to get the investment from all the different sectors to actually move the ball forward. So that's more just a comment. Thanks.
I mean, I think you're dead right, Matt. You might decide this is like a government, jargon or whatever, but this morning at 9:42, I got an email stating that there's a group that formed amongst the government side of the National Microbiome Initiative. It's the Microbiome Interagency Working Group. I mentioned them. Many of the people here at the table are members. And then I got an email this morning saying we never got our strategic plan approved by OSTP because there was a change in administration. But I just received an email saying that our charter has been extended to the end of December, which is great because that means it gives us more time potentially to actually get this plan approved. And the reason you should care is in order for us to work together, we actually have to have the charter. We have to have like permission, if you will, or the way paved. And I think now maybe we're closer to working together as agencies around this common need. So that's actually kind of a good thing that just happened today.
So I know that one of the passions of my boss, Dr. France Cordova, is public-private partnerships. And in Biological Sciences Directorate at NSF, we're really proud of those. We have a lot of them. The one that I'd like to talk about is the BREAD program that we do with the Bill and Melinda Gates Foundation, where we're partnering in sort of practical applications of relevant plant biology, agriculture, applied science for developing countries that are in distress with regard to food. But these partnerships, there's a lot of the house of no in government about making those work. And what I'm really pleased about is that More recently, there's a tendency to actually say, okay, let's figure out what we need to do to get those partnerships to work. Not only between government and private philanthropic foundations, but government and industry. So I'm actually cautiously optimistic that we may be turning the corner there.
Just wanted to echo some of the comments on the panel just in the last few minutes, actually. I lead the program our microbiome program at DNA Genotech. My name is Aaron Del Duca. And one role for industry potentially is in the creation and broad dissemination of tools. A lot of the academics and PIs aren't really funded to develop these things and get them broadly available, but that's a role that industry is very, very keen to play. And I'm really glad that we're having this dialogue now on that theme. Maybe a provocative question for the group of you, though. Do you think it's ready to elevate microbiome to the level of an all of us kind of study or an all of our bugs study?
Or like Mike Snyder was talking about a foodome project, which would sort of intersect there. I suspect we're going to go in that direction, certainly with all of us. And in fact, that will be the last question for the audience or anybody. I was asked if a large national research cohort was to be mounted. This is like all hypothetical. And they wanted to include a microbiome component. The question to me was, what sample should they collect? Because they're not sure what they would do with it yet, but they don't want to miss an opportunity to have a sample from the subjects to be able to do microbiome analyses. The first one that was posed to me was saliva, and I said no. I said, so even though saliva is easy, because depending on your age, your gender, what you ate, how dehydrated you are, how well you brush your teeth, if you have periodontal disease, that saliva sample is going to have a lot of noise in it. But they argued, well, it's easy. We're already planning to collect saliva. Can we just leverage a microbiome component? So if you were to mount, and anybody can answer this question, if you were to mount a national research cohort, and you wanted to hedge your bet and bank a body sample to be able to do microbiome analysis down the road, and I think all these agencies are thinking about this, what would be the most useful one? It sounds simple, but when you think about it, it gets very complicated. Anybody want to tackle that one? Well, let's start with humans. This is the human microbiome workshop. Everybody automatically say stool? Automatically say stool.
Yes.
But
And the reason is
go ahead,
because
please.
I just attended a steering committee meeting on Tuesday, Wednesday, and that was related with environmental influences on child health outcomes, which is a $165 million program by NIH per year for 7 years, and looking for the outcome of children who have either obesity, neurodevelopment problem, or any other kinds of problems. And looking for microbiome, the only sample which they said should be collected from prenatal as well as postnatal different life stages is the stool sample.
That was easier than I expected.
Hi, I study the oral microbiome, and I just want to maybe champion this saliva as being both a microbial sample and has serum and other biomarkers. And there's a lot of work already in using salivary samples as biomarkers for all kinds of diseases. So there's a lot of effort already put into that already. So and it's easy to collect. So anyway.
But if we had somebody here from NIDCR, like Dwayne Lunsford, he'd totally shoot that down. And that they responded just like you did.
Right. Yeah.
So I'll report back that we have a champion for saliva and apparently more champions for stool. I think I should wrap it up, if you don't mind. I'm not sure I'll be able to recollect everything that I heard, but I heard a lot about the need for more training, more trainees, more researchers in this field. I heard the need for trying to move to understanding the functional properties of the microbiome, and that would require deeper dives into the microbial membership through through understanding the individual members of the microbiome, through having genetic tests or genetic models for understanding these microbes. Help me recollect all the major themes that came out here. Modeling was a biggie, and that might be a real opportunity between NSF and NIH, because there's not a lot of modeling that happens at NIH, and I think the NIH community could benefit from modeling approaches. And talking about under the Kleiner model, there is a call for alternate models besides mouse or macaque. I think there was a lot of discussion yesterday about that. The need for standards. They need to be more inclusive in our cohort studies by gender or by racial and ethnic groups. Have I pretty much covered the main themes?
Can I add one thing, which I did not see in the presentations, that transparency or the reporting of the sex in the data, because only few presenters, they mentioned like what kind of sex they were using in their studies.
Yeah.
Like hepatocellular carcinoma person mentioned and a few other, but otherwise people are still not reporting. And as I have showed you in my presentation, there is a very big difference between males and females.
Mm-hmm. And I would agree with that, and I would also agree that maybe another gap that should be addressed is agreeing as a community to what we call a particular property, so we could compare across studies. That's a big charge too. I'm probably missing a big one. Anybody want to call it out? No? Well, thank you, everybody. Really, really appreciate it.